So please wait for 2-3 minutes. We will be starting soon.
Yeah, good morning.
Okay. So give me the sharing mic. Good morning, Dr. Prashant. So you will.
Yeah, good morning. Can you please share my presentation slides? Probably I'll be using this mobile phone.
Okay, yeah, no worries. So I will be sharing. So
Yeah,
Roshan, give me the
I'm
rights
using
for
mobile
sharing.
phone.
Yeah, no problem. So good morning, everyone. So let us start the proceedings, and I would like to introduce our first speaker for today, Dr. Prashant Shovajala. Dr. Prashant will deliver a talk on Introduction to Systems Genomics. Has obtained his PhD in Systems Biology from Aalborg University in Denmark. And he went on to gain more than 7 years of postdoctoral experience across 4 different laboratories. Prash worked for Birla Institute of Scientific Research, Jaipur, on his return to India, where he has successfully led 3 projects in the areas of systems genomics for rare diseases and next generation sequencing analysis of cancers and diabetes. He has interests exploring the known unknown regions in human genome, primarily working on the top-down systems biology approaches of hypothetical proteins. Over the last 8 years, he has developed an interest in long non-coding RNA in humans, elucidating the mechanisms underpinning small molecular interactions through clinical exomes. His group has benchmarked pipelines and developed methods for system genomics integrations. He is an associate editor of Frontiers in Genetics in the systems biology domain, and also bioprotocol, biomolecules, etc. BMC Medical Genomics, and also he advocates open access and open science for everybody. And he is very active researcher, and you will really learn a lot of things from his talk today. So I look forward and also welcome on stage Prashant. So now I will share the slides for you.
Yeah, please. Yeah. Thank you. Thank you so much, Pankaj, for your kind introduction and invitation. I apologize, for these technical issues that I was facing. I hope my voice is audible. Yeah.
Yes, you are perfectly audible. Just
Yeah.
let me share the slides now.
Yeah. Yeah. Thank you. Thank you so much. Again. So the next 1.5 hours probably we'll discuss about systems genomics, a fascinating field, that I've been associated over the last probably 1.5 decades. So if you really talk about systems genomics, or for that matter any interdisciplinary sciences, one of the striking features that really comes to mind is integration of machine learning approaches. So this particular cartoon at the extreme left side gives us a gist where we have a wonderful island here, and this girl is lost in an island, and as you could see, she don't know what to do, but she collects some pebble stones and she writes help with those pebble stones, but no one really comes to rescue. But she reimagines herself. She discovers that yes, why not, I mentioned something like machine learning, because she's a machine learner, and she writes and she collects few more pebble stones, and she writes, I know machine learning. And you see, how this wonderful, jet airplane also, comes to rescue. So this is the beauty of these, machine learning or integrated, life sciences. And that's how, or that's why probably all the IITs are really meant. And that is also certainly the need of the hour, post-COVID times. We need, collaborations, we need convergence, and we need, consistency, to the fore. Next slide, please. So I come from Amrita University. I'm really proud self saying that, Amrita University is one among the top 100 universities in the world, and we rank among the top 1. And this is a wonderful lush green campus in the Malabar southwest coast of Kerala in Kollam, where we have the main campus. We have got 6 other campuses. I've recently joined here as a principal scientist in September. Earlier, I was at Birla Institute of Scientific Research and worked for several other universities in US and Denmark for about 12 years. Next slide, please. So whenever we talk about genomics, one of the main entities that we really do is, whenever we say omics, omics means many or monies. Okay, whenever we say genomics, it's nothing but the study of genes or study of many number of genes. In my view, the most important facets of genomics is essential, not just because we have these particular data across different databases, but we owe something to someone whenever we talk about these particular entities. So I'm personally grateful to these 4 Gs. One, of course, gurus. It might be your own teachers, lecturers who have taught you, your spiritual gurus or whatever. The second one is Gandhiji. In fact, I'm a firm believer of openness. In fact, we owe a lot to Father of the Nation in many ways, because he was the first one probably who promulgated open access or probably open source, because everything that he has believed was given free to the world. And I think when we are celebrating 150 years of Mahatma last year, I think we owe a lot to him as well. And the third one, of course, genomes. It's also another G. That G is nothing but whatever data that we are trying to publish, it should be reproducible, and that reproducible data is available in the different databases. And that's how we pick all these the genomic reporter, and we should also be grateful to this particular genomes. And the fourth one is the Galaxy group of scientists. There is a wonderful cloud-based interface called Galaxy that was promulgated by James Taylor, who passed away last year, unfortunately. So he is a wonderful scientist. He's of my age as well, 42. By now he would have been 43, but he passed away unfortunately at a pretty young age. That's very unfortunate news, because he has given a lot of genomic data to the world. And that's how we did a plethora of systems genomic works, where our works have also been featured on these wonderful journals like Indian Journal of Cancer. Next slide, please. So whenever we talk about bioinformatics, I'm sure right from the wonderful speakers you have had during the very first 2 days, including Raghava and others, they might have taken to the wonderful prototypes of databases, bioinformatics, and all those kind of things. So whenever we talk about bioinformatics, we start with ensuring how these particular genes are integrated, how particular genomes fascinate us, how to view those particular genes across the genome browsers, how to compare the sequences. So for example, BLAST, we have where we can compare sets of sequences from one organism to the other organisms. We also talk about signature sequences. There might be some conserved sequences all through the evolutionary instances that could also be taken into consideration. There might be short signature sequences in the form of motifs as well that play a very important role. And most importantly, whenever we do genomics, the most important element is the mutations, or what we call it as single nucleotide polymorphisms. So these are all, the basic substitutions that we always, try to, bring to the fore. So whenever bioinformatics is really taught, we take the sequence information from the genomic reporter, we try to compare, then we try to predict the structures, and then, we try to give some kind of, function to these particular sequences. Okay. But through these particular processes, one of the most important elements that we always talk is, can we really compare one genome to the other genome at one go? Can we really simply compare and say that yes, you have sequenced one genome? Can we say that yes, this genome is just similar to this? Can we really say that yes, these genomes are similar? Can I ascribe function to that of that particular genome that is well annotated or curated? Okay. So for this, several databases have been produced. In case of primates or mammals, there is a HapMap project called Haplotype Mapping Project. In case of plants, there is something called Gramine. In case of bacteria, there is something called Bacteriome databases, and several databases, so on and so forth. Next slide, please. So but at the back of this mind, there's something called holy grailing, which means you're ascribing function to these sequences, as I was describing you. You have some sequence that starts with methionine. In case of a protein sequence, you have some structures that needs to be deciphered for those sequences, because sequence structure function relationship is the central dogma of bioinformatics. So imagine some set of pearl beads that are put up in a ball. You don't know what to do with those particular pearl beads, but if you insert a small thread in that, you can probably use those pearl beads in the form of a necklace, or in the form of an ornament. Okay, so likewise, all those nuts and bits and pieces of those particular protein amino acids are sewed together, are inserted together, and then you have folds. So when you have fold like this, you have those grooves, or the ligands to which it can really bind to that, and then you can say that, oh yes, this particular protein structure has got these ligands or these grooves, and as you can have some function ascribed to that. Okay, so sequence structure function relationship is absolutely very, very important in order to understand an orderliness of a protein. So if you have an order, then you can have a function to that. Okay, likewise, not many sequences do not have that kind of orderliness. Okay, so you have to say that yes, there might be misfolds. Okay, so whenever you have threads or daga, you have to remove some wild knots, which cannot be removed. So likewise, there are some wild knots that are misfolded in a structure, whenever you try to decipher a structure. So these are all the misfolds. So if you have misfolds, you may not really design or come up with a good function. Many a time, there are certain sequences which do not have structures as well. There are some 60,000 structures so far, and that is my discipline, because there are certain sequences that do not have a function. Those sequences are called as orphan genes or hypothetical proteins, and they have an utmost disorder. So there is no orderliness at all for those particular sequences. Then you cannot describe any function. The sequence unstructure unfunction is what we call it as disorderliness, and that's one of the reasons why systems biologists like you and me probably can play a very important role to ascribe function to these sequences. Next slide, please. Yeah. As I told you, there are about 60,000 structures. You come up with different sequence relationships, different databases, different resources. There are over 10 billion sequences that are publicly available right now. We need some kind of valuable information from these plethora of sequences. Next slide, please. So as of today, when I was just making or updating my slides, I mean there have been a plethora of sequences. As on today, until 9:16 hours, there have been more than 10 billion of sequences. If you can see that particular map, the red line indicates the whole genome sequencing data. The blue line indicates the genomic stuff. So it is drastically increasing because there have been, plethora of, genomic repertoire, that is flooded with all these particular databases. But this flooding is also on the premise of something what we call it as Moore's law. So if you are a computational scientist, you must be knowing what is a Moore's law. The computability or the computational capabilities would be doubled, with every 2 years. But within this particular doubling time, the cost of those particular computability would also be reduced to half, which means erstwhile you might be using a normal 2 GB or 4 GB RAM, but these days, unless otherwise you have a minimum of 16 GB RAM in your desktop computers or your laptops, you may not really use it. Okay, so that computability is also essential. So likewise, you have those particular huge data that is flooded with this genomic repertoire. Okay, now as this particular data is really flooded, the most important entity that you really ask is, can we really put this enormous data where, can we really afford such particular data into practice? Because the data storage would also, be, very much, important. Is it really, drastically reducing to half? So that is what we call it as, Moore's law. So it is in analogy with absolutely, the DNA sequences and the data. Next slide, please. So having said this, bioinformatics has really moved up the value chain. Okay, so earlier, bioinformatics सिर्फ एक tool जैसा कहलाते थे, not as really, as a subject. Okay, but its discipline like computational biology has really come up as an interdisciplinary sciences. Okay, where you do certain math or programming or statistics, biophysics, and all those kind of things. But in milieu, if you see, systems biology is such a wonderful discipline. You always talk about a system, be it a computing system or be it a vehicular system. Okay, where you have a car, all those car components are connected by different vehiculums, small components, and all those vehiculums are interconnected again with small nuts and bolts and pieces. Likewise, you have a system. Let's take a human system. So if you really look at me or you, we have body parts. These are all different body parts which make up major components. Okay. Likewise, every human organism has got about 1 trillion cells. Each of those trillion cells has got one nucleus, one mitochondria as a genetic material. If you really talk about the plants, you've got another chloroplast as another genetic material. Okay, and each of these particular organelles have got those particular proteins localized in them, and they keep interacting. And now, when we have the organisms per se that are very small, that are infecting us, something like SARS-CoV-2 here, okay, they are in fact infecting us. They're adapting to our environment, okay, and they're making different strains. Because erstwhile we had only alpha strain, beta strain, delta, gamma, epsilon, and now we have got something like Omicron, okay, which is some way alarming. So which means the viruses, even virus as a system, has adapted into a kind of an environment in such a way that it has known how to acclimatize to different conditions. Okay, so that is what we call it as strain adaptation, or strains adapting or unfitting in the environment. So you got mutations arising and falling in an environment within different conditions. Maybe you might be in somewhere in Jaipur where you might be acclimatized to pretty what you call hot conditions. I'm also acclimatized to hot conditions because I was born in India, but my presence in US and probably Denmark, where I guzzled minus 20 or minus 30, I might have acclimatized some sub-frozen temperatures as well. So maybe there might be genetic variation that is certainly be associated between the persons who are living in, say, extremities, somewhere in Arctic region, or somewhere in the Antarctic region, or somewhere in the humid or temperate conditions. So these influence a lot on how a genome is exploited. So I would say that systems biology was lost into oblivion once. Maybe it was existing at some point of time, but as strains adapt, as genes are lost and found, as proteins evolve and devolve, as mutations arise and fall, maybe this systems biology has really transcended into systems sciences or systems genomics. Okay. So the take-home message from this particular slide is interactions play a very, very important role. Okay. Next slide, please. So when I say environment plays a very important role, or interactions play a very important role, environment is the bottom line, okay? Because environment plays a crucial role. And midst of this, you have got some context, okay? There are some contexts in the environment, you talk about, say, plants, you talk about stress traits, the quantitative trait loci, the genes. You talk about bacteria, you talk whether or not that particular bacteria is a monoisolate or multi-isolate, or it is pathogenic, non-pathogenic, host specific, host non-specific, all those kind of things. Likewise, you talk about animals, whether or not their feeding efficiency is quite good. Okay, all those things that you talk. And whenever you talk about humans, you've got different variables, different diseased phenotypes. Some persons might be associated with different diseases, different epigenetic environment, and all those kind of things. And the common variable is environment. Okay, so that is a very, very important thing. So, and that's how you can summate this up to, you might have certain true positives, true negatives, false positives, and false negatives. Okay, so next slide, please. Yeah. So this is about systems genomics. You need a multidisciplinary pathways. This is one of our highly cited articles, which we use multi-omics approach, multi-omics integration for this. So we need genomic data, we need a proteomic data, we need an interactomic data, we need some data that is coming from the non-coding chunks, and they need to be assembled together, and then we need to really validate and come up with a multi-omic integration, which plays a very, very important role. Next slide, please. Yeah. So so there are some user friendly tools. As I told you in the very first few slides, Galaxy project plays a very important role. Galaxyproject.org or use galaxy.org. Simply आप आपका genome upload कर लीजिए. You use some tools. You pick which tools you need to drag and drop, and it will do it for you. Okay, that's a wonderful tool that is produced by Galaxy Foundation. Likewise, Princeton has developed wonderful tools like JANT, IMP, and Seek. There's also something called BioGrade, Genome Space, and CBio Portal, which have been used, for different things. Next slide, please. So a quick summary of my, first part of my talk here is systems genomic approaches play a very, very important role. We need, multifaceted disciplinarians to come together. We need functional annotation approaches. We need, consortia to be brought. We need, multidisciplinary collaborations, that need to be converged, not the competition. Collaborations are absolute, key for success for bringing, systems genomic approaches. And most importantly, if you find a gene of interest, if you find a protein of interest, you need to ascribe function to that. And for that, you need a rigorous curation. And we need, different bioinformatics pipelines. And for that, we need to dig deeper. What exactly, that system is really doing? What kind of, genes are present in that particular system, say human system or, bacterial system or whatever. Okay. And for that, you need, next generation sequencing approaches. Okay. What is that? We'll go through the next slide. Yeah. So this is where the next generation sequencing has come into picture. So the very first part, we were talking about why system is needed, how system is capable to ascribe some kind of function, but why and how we need to ascribe a function. What kind of mutations that we really need to understand. That's where the next generation sequencing has really come into picture. And it is also called as now generation sequencing, because next generation sequencing was coined way back in 2009 or 8, but now it's been almost more than a decade, and we need really some kind of things. Next slide, please. So you might be, maybe I'll just quickly pause here. If someone has some questions, I really want this to be interactive. Yeah. If you don't have any questions, maybe I can continue. Yeah. Yeah. So Pankaj, can I continue? Assuming that there are no questions. Yeah.
Let me see if there are any questions in the chat box. No, not really. So yeah, you can continue maybe. Yes.
So you might be knowing what is Sanger sequencing. The erstwhile Sanger sequencing with the Human Genome Project was sequenced, some 25, 30 years ago. There's something called dideoxyribonucleic acid sequencing approach. So you get some signals, whenever there is an A, T, G, and C, and sequencing a small bases of 50 bases, used to take, hours actually. But in 2009, thanks to Illumina sequencing strategy, there have been, wonderful approaches. So there have been a completely big change on how you really look at the sequencing strategies. So if you really see here in the slide, you have got a DNA strand. Okay, DNA is split into several bits and pieces. Okay, I'm not sure if you can see my screen. So imagine this particular DNA like this. Okay, like this yellow paper is a DNA. Okay, it is split into several bits and pieces, and then what you do is you try to add something called adapters at the end. Okay, these adapters are synthetic bases like this. Okay, and now these adapters go through the length and breadth of these particular fragments. So each of these particular sequences, okay, or fragments are split into several times. Okay, several reads are oriented, and you get these particular adapters, that are, ligated at the end. Okay, now what happens is there is something called coverage, okay, which I'll probably describe at a later point of time. But these adapters are synthetic bases. So each of these library preparation steps has got, different adapters. There are also linkers that are adhered, somewhere every 10 or every 20 bases in between. And now these are hybridized. You design some PCR primers, you amplify them, then you enrich the libraries, okay? And after the enrichment and the amplification is done, you sequence, you put them in the instrument, and then you get these A's, T's, C's, and G's. Okay. Next slide, please. And then you might ask me, what is enrichment? Okay. So in the enrichment, you can use different PCR chemistries, different inversion probes. Okay. Different direct capture genomic selection based approaches. Okay. And then you talk about the number of read lengths that you really want to ascertain, and it all depends from one particular sequencing platform to the other. And Illumina is that sequencing platform, which is a short read sequencing chemistry, which uses anywhere between minimum 50 bases to 500 bases, whereas long read sequencing like PacBio or something like Minion, okay, Oxford Nanopore technology. So this is Oxford Nanopore technology. I have this sequencer with me, okay, here. So this uses, anywhere between, I would say, minimum of, 10K bases to maximum, 50K bases or even upper, okay. So different, sequencing platforms are used, which we'll discuss, next slides. Next, please. And then you get some raw reads. Raw reads are obtained in the form of something called FASTQ files. In case of Sanger sequencing chemistry, during those days, used to get something called FASTA format. But now FASTQ is fast quality. So each of these FASTQ files are obtained in the form of raw reads, and then you map it to your reference. Okay, whatever reference that you have, you need to map it so that you need to see how much alignment sequences are aligned to your reference. If you don't have a reference, you can simply do something called de novo strategy, which means it is a kind of an ab initio based approach, which you assume that, which you ask the program and say that yes, it is your methodology that you really want to employ, and then map it to your proprietary databases at a later point of time. Okay, and then you go through different things. You convert your file to sequence alignment mapping, then binary alignment mapping, because BAM files are preferentially used because it reduces the size of your file. And finally, you get the VCF file, the variant calling format in case of whole exome sequencing and whole genome sequencing. Okay, and then you map it to these databases, the proprietary databases that are available, and then you filter the variants and identify what are those variants, that are pathogenic in nature. Next slide, please. There are different databases and pipelines. Well-known commercial pipelines like Multi-Omics, Partech, and other things do exist. For whole exome sequencing and for whole genome sequencing, different pipelines like Genome Analysis Toolkit do exist. And there are cloud interfaces like Galaxy, which I was telling you, and Taverna and WebEx also are used. But at the end of the day, you need to reach consensus. In bioinformatics, you always need to reach consensus. You cannot say that, yes, you have used one tool, ठीक है, मुझे एक tool से ये particular result मिल गया, and hip hip hooray, you cannot say that. Okay, you always need to reach consensus using one or two more pipelines or tools, and then you can come to a conclusion. Next slide, please. So this is what I was telling you. You have FASTQ file, you have something called, you need to convert to SAM, BAM, and finally you need to get a VCF file in case of whole exome sequencing and whole genome sequencing. What is that? I'll try to tell you. Okay, but you need to tweak your tools sometimes. Kabhi kabhi, if you are a bioinformaticist, kabhi kabhi kuch different options use karke, you have to use it. Okay, that's what we call it as, benchmarking the pipeline. Okay, next slide, please. So what happens is, when your DNA is present in your nucleus, the chromatin is uncoiled. That's split into equal sized fragments of, say, the ideal size is 150 base space in case of short read sequences or Illumina sequencing chemistry. So each of these 150 reads are oriented like this, and the adapters are put up like this, if you can see my screen. Okay, and now depending on the orientation, which means if the sequencing read is just oriented from left to right like this, it is what we call it as a single end sequencing chemistry. If the read is oriented from forward and as well as backward or reverse, that is what we call it as a paired end read chemistry. Many a time, you need to identify or amplify multiple number of single end reads or multiple number of forward reads and multiple number of reverse reads. That is what we call it as mate pair chemistry. Okay, and the most important thing is, once you get these particular sequencing library done, you need to quality check. Is base calling done properly or not? Are you getting all the bases promptly or not? Or if there is no ATGC, are you getting N? N is noise. Are you getting any kind of noise? Okay, that is what we call it as quality check. And after this is done, the most important thing is you need to remove these adapters, because these are synthetic bases. These simply act as something like barcodes, isn't it? So now you're removing it and you are obtaining your real sequence again back to your life. This is where you need to really compare it to your genome of your interest. Okay, is it clear? Next slide, please. Yeah. And then you might ask me, Presh, ठीक है, आप तो sequencing library के बारे में आप बताएं. You are talking about the forward chemistry, the reverse chemistry, the type of sequencing platforms and all. Then how do I know what type of sequencing that I need to do? That is a very important question. To me, that's a very striking balance that you need to ask. Whole exome sequencing is an ideal one if you are looking into a diagnostic front. मान के चलिए आपके पास एक organism of interest है, like human disease phenotype. You want to identify the mutations in those particular patients, then as a diagnostic front, whole exome sequencing is the best. Here you only chop off the exons. Okay, so if you have a human genome of say 3.2 billion base space for a haploid data set, for a diploid data set, it is 6.4 billion base space, right? So 1.8% of the coding chunks are nothing but the exons. So 1.8% of 6.4 GB of data roughly translates to about 58 MB. Okay, so मतलब 58 MB of exons are present in your genome. उन सारे 58 MB को अगर आप target करेंगे, then that is nothing but whole exome sequencing. Whereas whole genome sequencing is entire stuff. Okay, you are sequencing both introns and exons, but it might prove to be very costly because आपको libraries, आपको पूरा intronic chunks को भी पूरा निकालना होगा. मतलब 3.2 billion base pairs of the entire haploid data set को आपको करना होगा. But your lab may not be rich. You know that you have some known mutations that are already identified, but you want to go for only targeted chemistry. मतलब आपको यदि पता है कि क्रोमोसोम 1 इस पर्टिकुलर रीजन में इन सारे म्यूटेशंस हैं और BRCA जीन में इन पर्टिकुलर म्यूटेशंस को ही आइडेंटिफाई करना है, then you can do something like targeted genome sequencing, which we call it as TGS. Okay, your sequencing chemistry might be something different. आपको सिर्फ ये नहीं है, आपको बल्कि अभी तक होल एक्सोम टारगेटेड जीनोम होल जीनोम सीक्वेंसिंग में सिर्फ आप क्या है डीएनए की बात कर रहे हैं, डीएनए एज ए सीड मटेरियल. बट आपको एक्सप्रेशन यदि निकालना हो, ओके, then you use something called whole transcriptome shotgun sequencing, okay, or RNA-seq. That plays a very very important role. Okay, तो इसमें क्या है, you take RNA as a seed material, you reverse transcript to cDNA, and that cDNA expression profiles are measured, and then you compare it with your wild type. How many differential expressed genes are known? Maybe if you're a bacteriologist, if you're a biologist, or if you're an environmental biologist, you want to do a metagenomic sample. Okay, metagenomics, meta means many, genomes is multiple genomes. So if you collect some samples from an environment or from a source, you don't know what kind of organisms do exist in them. Okay, so it might have a lot of bacteria, it might have a lot of microeukaryotes, small fungi, and all of them. So you may have to do, metagenomic approach for that. Okay, that's how your sequencing is. Next slide, please. So these are the statistics which I was just telling you. Imagine you are somewhere 10 kilometers above IIT Jodhpur. Okay, आपको कुछ भी नहीं दिखाई देगा, आप पैराग्लाइड कर रहे हैं. Okay, maybe you want to jot down to IIT Jodhpur Department of Biosciences somewhere, over there. So if you really want to zoom in and drop somewhere over there in Department of Biosciences, it is going to be very difficult, isn't it? It's not an easy thing, coming from 10 kilometers above the sea level. You cannot see anything, but somewhere 2 kilometers, 500 meters तक आने के बाद आपको IIT campus आपको दिखाई देगा. But you will not get down exactly at Department of Biosciences, okay, in IIT Jodhpur. You might fall short of that, building somewhere, adjacent to that. Likewise, भले आप coverage आप लेंगे, okay, मतलब if you want to identify or sequence these particular fragments 100 number of times, 100x coverage अगर आप यदि बोल रहे हो, आप 100x finite goal you'll not get that. You might fall short of it. You might get up to 90x coverage, depth of coverage, or 80x. Okay, not all fragments are sequenced impromptu. Okay, so it all depends on the kind of sequencing chemistry that you are trying to ask. Okay, so the most important thing that you really want to see is whether or not your target sequencing chemistry is achievable or not. Whether or not your sequencing यदि आप कर रहे हो, आपके पास एक organism of interest as a reference है कि नहीं. यदि है, you can map it to your reference and see how much alignment is seen. If you don't have, then you can simply use something called de novo strategy. Okay, so this is what I was just telling you. In a whole exome sequencing strategy, 1.8% of coding chunks of 3.2 billion base pairs of human genome for a haploid data set roughly translates to 56 MB for a diploid data set. Okay, अगर यदि आप 100x coverage पे अगर आप sequencing करेंगे, then that roughly translates to 6 GB of your data per sample. Okay, देखिए, अगर आप paid end sequencing कर रहे हो तो 6 GB for forward read and 6 GB for your reverse read आपको मिलेगा. ठीक है, मतलब 12 GB of your data यदि आपको analyze करना है तो downstream analysis में 12 गुना 5 मतलब 60 GB of data आपके downstream files occupy करेंगे. उतना space आपके पास computability के लिए होनी चाहिए. That kind of Moore's law जो मैं आपको बता रहा था, उतना computing space, उतना RAM, maybe 1 or 2 TB RAM minimum, like what we have, you must definitely have that. Okay, so these are the rough statistics that you really need to know. Okay, Next slide, please. So there's a subtle difference between whole exome sequencing and clinical exome sequencing. In a whole exome sequencing, you chop off the entire 56 MB region and identify the mutations. In a clinical exome sequencing, सिर्फ आपके पास एक disease है, disease में क्या क्या mutations होगा, आपको पता होगा, सिर्फ वही mutations का आपको निकालना होगा. And the turnaround time is anywhere between 14 to 16 days. बट आप जैसे और मेरे जैसे अच्छे बायोइन्फॉर्मेटिक्स और सिस्टम्स बायोलॉजिस्ट है तो हम सिर्फ 2 या 3 दिन में हम निकाल सकते हैं. ओके, इफ यू हैव गुड पाइपलाइंस. नेक्स्ट स्लाइड प्लीज. सो एग्जाम्स में एक छोटा सा डिसएडवांटेज भी है. यू कैन नॉट आइडेंटिफाई ऑफ कोर्स म्यूटेशन सिटिंग इन द नॉन कोडिंग चंक्स, है कि नहीं? बिकॉज आप सिर्फ एक्सॉन्स ही आप चॉप ऑफ कर रहे हो और लाइब्रेरीज आप सिर्फ एक्सॉन्स के लिए आप निकाल रहे हो. But कई मर्तबा ऐसा होता है कि genetic variation is best seen in the non-coding RNAs as well. You might find mutations that are represented in the non-coding chunks. Okay, so those particular things, those structural variants like copy number variants, they might be missing, from your exome sequencing chemistry. Next slide, please. So usually whole exome या whole genome sequencing में आप या even whole transcriptome में भी, हालांकि आप whole transcriptome और RNA-seq में आप भले जीन एक्सप्रेशन ही आप निकाल रहे हो, उसके बाद आप म्यूटेशंस भी आइडेंटिफाई कर सकते हैं, जैसे हम कर रहे हैं. हमारा एक प्रोजेक्ट है BST का, वी आर डूइंग दैट. Okay, very rarity, because the end goal here is only to identify the gene expression. Okay, how many genes are upregulated or how many genes are downregulated. But after that, you can see how many mutations are per se seen in those particular samples. Okay. Likewise, you have to identify whether or not a mutation is benign, which means it's not pathogenic, it's well defined, or malignant. Agar malignant mutation hai, to it's pathogenic. If it is disease associated or it is causal or bona fide, you can identify using this whole exome sequencing or whole genome sequencing. And that's one of the reasons why whole exome sequencing has been a big hit. It's cost effective. Over 200 genes have been discovered so far with varied mutations across different samples. But again, as I told you, the fallacy is genetic variation non-coding RNAs में यदि अगर आप निकालना चाहते हो, then you might miss if you do whole exome sequencing chemistry. But whole genome sequencing में you can do that. But whole genome sequencing करना बहुत मेहनत की बात है. I don't say मेहनत, but if your lab is rich, you can do it. Because उतना पूरा 3.2 बिलियन बेस स्पेस का अगर आप निकालना हो तो आपके पास 320 GB ऑफ डेटा आपको मिलेगा. हालांकि आप 100x कवरेज निकालने की जरूरत नहीं है. यू कैन कम डाउन टू 40x और 35x कवरेज फॉर होल जीनोम सीक्वेंसिंग एज वेल. नेक्स्ट स्लाइड प्लीज. So this is what we have done. We have developed our own pipelines. Our pipeline was downloaded over 30,000 times for whole exome sequencing. It's available in bioprotocol. You give a raw data like this, a whole exome sequencing, small FASTQ file, and you'll get the final list of mutations, in the form of VCF file, and you can prioritize the variants. And this prioritization of variants is very, very crucial for systems genomic approaches, because you need to understand why or which kind of variants are very, very important or detrimental to your diseased phenotype. Okay, next slide, please. Likewise, whole transcriptome shotgun sequencing RNA-seq का भी हम एक कफलिंग pipeline हम develop किए, benchmark किए. I wouldn't say develop, it's a benchmark pipeline. You take your FASTQ files and finally आपको जितने भी differential expressed genes हैं वो आप निकाल सकते हो. Okay, and then you can validate them using qRT-PCR. For whole exome sequencing mutations, you can use Sanger validation. For differential expressed genes, you can do a qRT-PCR validation. Okay, next slide, please. So the learning points, the quick learning points for these particular things here is, Illumina is a short read sequencing chemistry. Over 80% of the users use Illumina short read sequencing chemistry. PacBio, ONT Minion, 10x Genomics are long read sequencing chemistry. There are some fallacies, although you can sequence larger fragments of anywhere between 10 to 50 kbps. But उसमें high error rate होगा naturally. Maybe imagine you are asked to pick up apples that are present in a row, in a big table, and you're asked to run from one end to the other end and pick up as many apples as you could. अगर 5 apples है तो you can easily pick up those 5 apples. But if you have thousands of apples in a big hall, and if you are asked to run and pick up all those apples, the instrument also falters because it's a long read, isn't it? So there are some fallacies, with these sequencing platforms and sequencing chemistries as well. Okay. Yeah. Next slide, please. Yeah. I'll quickly pause here. If you have any quick questions, I'll go through the case studies of this after these learning points. Yeah. Any questions, please? Yeah.
So I request the attendees to write their questions. If you have any question, feel free to write in the chat box, please.
So I know, maybe if you are all from Jaipur, I could imagine I was in Jaipur until August. It might be, bittery cold, but I'm experiencing, wonderful humid or summer temperatures in Kerala. I know bittery cold mein questions pucha nahi jata hai. I know. So please don't worry. You can ask me at the end of the session as well. Yeah. So the learning point here is, which instrument to opt for sequence short read sequencing. The best is, Illumina, as I told you. The latest technology है NextSeq 2000 और अभी 2500 अभी 3000 भी instrument अभी आ रहा है. You can do all these kind of things, like large genome sequencing, short read sequencing, metagenomic, transcriptomic profiling, single cell technologies, cell-free sequencing, 16S, methylation, MedIpSeq, ChIP-seq, everything, whatever you want. And the turnaround time is also quick, but you have to shell out a lot of, monies. But if you have more number of samples, it's always nice. Okay. Yeah. So the next slide, please. So so basically, next generation sequencing is used, to sequence plethora of genomes, be it microbiome, be it a human genome, be it identify the gene expression profiles or identify novel mutations or pathogens. Okay. And at the end of the day, you can also compare it with your genotyping as well. Okay. In fact, genotyping was prelude to NGS, the genome wide association studies. Okay. उसमें क्या है, सिर्फ आप एक बार you simply genotype, what is that particular specific base at a given position. Okay. And then say, yes, you have maybe this mutation for this disease phenotype, and that is how, you identify the mutation across the genomes. So that is a genome wide association based studies. But next generation sequencing may you can do quite a lot. Next, please. So this is about the orientation. You can do the paired end reads and mate paired end reads. As I told you, single read chemistry में सिर्फ एक बार from left to right you simply do, that is single end chemistry. But it's always do paired end sequencing to reach consensus. मतलब okay, the paired end chemistry is also very much important. And then you can also do whole genome sequencing. Next slide, please. Okay, but whole genome sequencing would cost quite a lot of money and time and energy. It costs billions if you want to sequence at least 100 genomes, because if you want to do some comparative based approaches as well. Next slide, please. Yeah. So you might want to ask me, Prash, आप कह रहे कि whole exome sequencing या whole genome sequencing या target genome sequencing करते हैं तो Sanger sequencing से validate करना होगा वो mutation. तो यदि अगर आप RNA-seq करते हो तो RNA-seq और microarray या qRT-PCR में क्या अंतर है आप पूछ सकते हो. Okay. Assuming that everyone here are renowned faculty, I think must knowing what does a qRT-PCR does, right? So it gives you the threshold cycles. It tells you the influence of number of cycles at which those particular fragments are amplified, and based on the amplification, you can know the what is the threshold at which that particular amplified fragment is seen. Okay. आजकल qRT-PCR जैसे बड़े-बड़े रॉश machines में आप multiplex भी कर सकते हो, which means 386 weld plate में भी आप multiple number of samples, multiple number of genes भी आप कर सकते हो. बट एक सैंपल में कई जीन्स यदि आपको करना हो, you might really falter, okay, with a multiplexing. You cannot do that kind of an expression using QRT-PCR. And then you do microarray. Microarray is sensitive because you have those fixed probes, you know, those generic probes that are known across thousands of genes. But उसमें भी आप multiple samples यदि करना हो, you might really falter. But RNA-seq is a wonderful strategy. You don't have such, you know, compliance. Okay, मतलब आपको you need not really have such kind of problems, you know, that you have here. Okay, इसमें क्या है, आप कई number of samples, कई number of genes across, you know, different levels पे आप validate कर सकते हो, and you can identify whether or not those genes are upregulated and downregulated when compared to your housekeeping genes. Okay, मतलब आपके पास एक control sample होगा, आपके पास एक disease sample होगा. Now your end goal is to identify what are those differential expressed genes between them. Okay, then you do RNA-seq as an end, but you need to validate them. Again, validation के लिए आप सिर्फ again RNA-seq use नहीं कर सकते हैं. QRT-PCR, some small number of, you know, sample validation of QRT-PCR से आप definitely आप कर सकते हैं. Okay, next slide please. So again, validation for whole exome or whole genome sequencing. There are different approaches, as I told you. Sanger validation is the best. You can use mass spectrometry based validation like CRISPR technology as well. Okay, next slide please. So Illumina has really gone through wonderful things. Matlab, Illumina has given a short read sequencing chemistry, which is highly sensitive, cost effective, high turnaround time, right from the good old, you know, CMOS technology जो computers में complex metal oxide semiconductor एक chip होता है, वहां से लेके उन लोग sequencing platforms, you know, generate करते आ रहे थे. और अभी अगर आप देखेंगे तो even 6 terabyte data तक एक दिन में, you know, generate करने का, you know, आवश्यकता है, you know, if you use Illumina short read sequencing chemistry. Okay, whereas long read sequencing ठीक है, results तुरंत दे देता है, एक ही बार देता होगा, but you might find some errors, you know, when you long read pooling. So the next slide please. Yeah. So the primary take home message here is you need to identify what to do if you have a reference. If you don't have a reference, you need to do de novo. What is whole exome sequencing? What is whole genome? What is targeted genome sequencing? For diagnostic front, we use whole exome sequencing. There are of course some tools like Bouty and ISAC, which I fairly missed telling you. In fact, those two tools are used, you know, to align your FASTQ files to your reference. Okay, and then identify, you know, कितना alignment हुआ आपके reference के साथ. Raw reads है, आपके पास FASTQ file होगा. Tools like SAM tools are used to convert SAM to BAM, and then you get the mutations. Okay, or cufflinks pipeline is used, you know, for RNA-seq, you know, which we have used, you know, for pipeline. Next slide, please. I'll quickly pause here. I have some case studies since it's a faculty development program. यदि आपके पास some problems है, तो मेरे साथ आप discuss कर सकते हो last 5-10 minutes. But I'll give you my problems, the wonderful 4 or 5 problems which I want to discuss over the last 10 years, right from my Denmark and US days. How did we overcome this? I'll probably discuss. I'll quickly pause here for 30 seconds. If you have any questions, yeah, I'll be happy to answer. Yeah. So assuming that there are no questions, I'll move forward. तो जब मैं इंडिया आ गया था 2016 में बिरला इंस्टीट्यूट ऑफ साइंटिफिक रिसर्च जयपुर, I just started my group in 2016 and I was you know, excited to do a lot of things. मुझे एक प्रॉब्लम दिया गया था. ये प्रॉब्लम ये था, नेक्स्ट स्लाइड प्लीज. या कि एक कंजेनिटल पाउच कुलन करके एक बहुत रेयर डिसीज है जोधपुर में, यू नो, उस इलाके में नो बहुत you know, फैला हुआ है. It's a rare disease. The child is born with a, you know, dilated pouch. इतना pouch होता है पेट में. कोलन इंटरकॉइल्ड है, तो लार्ज फिस्टुला की वजह से एक्सक्रीटरी system is also very bad. इस pouch को निकालना पड़ता है, but इसका अंतर क्या है, इसका reason क्या है, ऐसे बच्चे क्यों ऐसे जन्म ले रहे अभी तक पता नहीं था. So Dr. Praveen Mathur and Dr. Krishmon Merichella, who was my mentor. Dr. Praveen Mathur is a renowned pediatrician from SMS Medical College. He has given us this problem. तो congenital pouch colon ये था. Next slide please. तो मुझे लगा कि जब whole exome sequencing analyze कर रहा था, सारे mutations हम निकाल दिए थे, and then interestingly इसमें कुछ mutations ऐसा था कि non-coding RNA पे भी हमें मिला था. तो तब हमें डाउट आया था, अरे हाउ कम है होल एक्सोम सीक्वेंसिंग में non-coding RNA mutations कैसे मिल सकता है? This is what we tried to figure it out. Okay, next slide please. So what we did was, before which I'll tell you what is non-coding RNAs. Non-coding RNAs are nothing but those that are coming from the intronic chunks. They do not code for proteins. But halaki, you find certain genetic variation that is seen rampantly in those non-coding RNAs that is detrimental to the diseased phenotype. Okay, in other words, यदि आपके पास एक म्यूटेशन यदि हो जो नॉन कोडिंग आरएनए में है, वो म्यूटेशन ज्यादा डिसीज फैलने का संभावना है than a mutation that is sitting in the coding chunk. Okay, तो जब हम ये कर रहे थे, ये पर्टिकुलर म्यूटेशंस जब हम निकाल रहे थे, नेक्स्ट स्लाइड प्लीज, we found one non-coding RNA mutation which is non-exact 008982 non-human Homo sapiens transcript. Okay, वो म्यूटेशन हमें आइडेंटिफाई किए थे वो जीन में, बट हमें पहले हम हैरान हो गए थे कि हाउ कम होल एक्सोम सीक्वेंसिंग केमिस्ट्री में नॉन कोडिंग आरएनए म्यूटेशन आ गया. तो क्या हुआ होगा मैं आपको बताऊंगा. सो ये आप जानते होंगे, अभी देखिए ये पिंक वाला पूरा मान के चलिए इट इज exon and the yellow one is an intron. Okay, in this slide if you can look at. तो सीक्वेंसिंग लाइब्रेरी करते समय यूजुअली एक्सन्स को ही लाइब्रेरी करना होगा, मतलब यही आना चाहिए आपके पास. ओके, हालांकि सीक्वेंसिंग लाइब्रेरी करते समय आपके पास ये भी आ गया ऐसे, मतलब कुछ इंट्रॉनिक चंक भी आ गया. ओके, तो यहाँ जो लास्ट चंक भी रह गया था लाइब्रेरी प्रिपरेशन जब करते समय, तो यहाँ भी म्यूटेशन, यूजुअली वो म्यूटेशन है कि नहीं हम आइडेंटिफाई करना भी बहुत इंपॉर्टेंट है. बट लकीली the mutation that is just very close to the exonic chunk that at the intron boundary was found to be very rampant and very detrimental to congenital pouch colon. That is what we discovered, and we feel very happy that that's one of that's not one, it's the first such report where non-coding RNA mutations whole exome sequencing chemistry से हम किए थे. और ये चीज हम rather long non-coding RNA protein interactions भी किए थे using microscale thermophoresis, which is a biophysical instrument. You need not do a co-immunoprecipitation. So if you have a MST instrument, a nanotemper technology like microscale thermophoresis, इसमें आप long non-coding RNA निकालो और प्रोटीन निकालो प्योर फॉर्म में, और वो इंस्ट्रूमेंट ऑटोमेटिकली बता दे कि ये दोनों इंटरेक्ट हो रहेंगे. Okay, next slide please. So this is what we did. So RNA tissue हम nanodrops से भी हम किए थे. Next slide please. And then we have also isolated that particular KIF13A protein that is responsible for the disease. Okay, next slide please. And then we used some bioinformatics approaches, so long RNA with those KIF13A जो disease causing gene के साथ interact हो रहा है वो हम identify किए थे. Next slide please. And extreme right side में 12 to 3 o'clock position में जो sigmoidal curve आपको दिखाई दे रहा है, ये है माइक्रोस्केल थर्मोफोरेसिस, ऐसे रिजल्ट आता है आपको. ओके, इसमें वंडरफुल डॉट्स हैं. ठीक है, दोस डॉट्स एट वन साइड आर लॉन्ग non-coding RNAs एंड द अदर साइड आर ऑल प्रोटीन्स. ओके, यदि सिग्मोइडल कर्व के भीतर है इसका मतलब है ये दोनों इंटरेक्ट होने का चांस है. सो दिस इज अ बायोफिजिकल बेस्ड अप्रोच बेस्ड ऑन योर स्कोर. नेक्स्ट स्लाइड प्लीज. एंड वी पब्लिश्ड इट इन बायोमोलिक्यूल्स व्हिच वाज द फर्स्ट सच काइंड ऑफ रिपोर्ट, which was pretty fascinating. Next slide please. So the take home message here is whole exome sequencing का मतलब सिर्फ mutations from the exome sequencing ही exon से होना अभी अनिवार्य नहीं है. Okay, you can identify the mutations also from the non-coding chunks as well, provided you ask the librarian to identify the mutations from the non-coding chunks as well. Okay, so preferably a kind of a myth was busted. Okay, तो हम क्या किए थे, वो लॉन्ग नॉन कोडिंग आरएनए म्यूटेशंस और वो जीन्स है जो किफ 13A के साथ, उसके साथ हम आइडेंटिफाई जो हम किए थे, हम कंजेनिटल पाउच कोलन के रिलेशन हम निकाले थे. Okay, and this is the first microscale thermophotosynthesis study in rare diseases, at least, which we have shown in congenital pouch colon. Okay, this is a wonderful systems genomic approach, which we try to do that. Next slide, please. And then I was always interested in prostate cancer right from my postdoc days, because मैं 7.5-8 साल postdoc कर चुका हूँ. उसमें जब मैं एनिमल जेनेटिक्स लैब में मैं कर रहा था, काउस एंड बफेलोस बेचारे ग्रेस करते समय बहुत ग्रोन करते थे, बहुत आवाज करते. तो मैं बेचारे क्यों कर रहे थे, इज इट बिकॉज ऑफ ओवर ईटिंग और व्हाट, वी डिडंट रियली नो. बट जब हम वेट से जब हम पूछे थे, उनका कहना था कि इन एनिमल्स भी बेचारे बुढ़ापे में 50 इयर्स के बाद, ह्यूमन के 50 इयर्स के बाद, मतलब काउ के 20 और 30 इयर्स के बाद, उनका प्रोस्टेट ग्लैंड भी बहुत एनलार्ज एंड प्रोस्टेट कैंसर आने का चांस है. तो मैं ह्यूमन प्रोस्टेट कैंसर में भी मैं बहुत इंटरेस्टेड था. तो जब इंडिया आया था, आई वाज आस्किंग if there is any prostate cancer exam data that is known. Okay, fortunately no one has done, unfortunately no one has done, I don't say fortunately, but no one has done that. So we started it. Next slide, please. So prostate cancer क्यों? Because 1 in 9 men are diagnosed with prostate cancer during the lifetime. India में भी अभी 3rd prostate cancer है, and fertility rates and androgen receptor signals are also lost in those particular population as well. Next slide, please. So what we did was we collected some samples. Prostate cancer Rukmini Birla Hospital Jaipur से हमारे collaborators हैं. What we did was we have taken different Gleason grades. Gleason score above 5 इसका मतलब है वो prostate cancer victim है. उनका prostate cancer tissue हम निकाले थे. Benign prostatic aplasia is a normal sample. Gleason score less than 5. Okay. और इनमें what are the kind of mutations है हम identify करने के लिए हम try किए. Okay. Next slide, please. And to our utter surprise, usually prostate cancer meat eating people में आता है, non-vegetarians में हालांकि आता है. That's the reason why US और African population में बहुत ज़्यादा है. But हम ज़्यादातर लोग हम घास खाने वाले हैं, जो भी Indians so called, almost 63% of Indians are vegetarians. अगर आप जयपुर का अगर आप देखेंगे, राजस्थान में तो almost 85% are vegetarians. And yet जो हम samples collect किए थे, initially 10 samples में 9 patients were all vegetarians, and yet they had this prostate cancer symptoms. We were surprised. और एक और बंदा भी ऐसा था कि is not really he's non vegetarian. वो अंकल जी भी थोड़ा बहुत एग खाते थे. एग हालांकि it's a lacto vegetarian हम मान के जा सकते. Okay, so we were surprised. So we asked, you see here, we found mutations linked to obesity, vitamin D, mitochondria, and vegetarianism as well. मतलब diet related things as well. तो ये सारा जो सर्कुलर म्यूटेशंस हैं, सर्कुलर जीन्स है, वो सिर्फ इंडियन में दिखाई दे रहा है, और स्क्वायर वाले हैं सिर्फ यूएस और कॉकेशियन पॉपुलेशन में दिखाई दे रहा है. एंड वी फाउंड सम नोवेल म्यूटेशंस दैट आर स्पेसिफिक टू इंडियन कंडीशंस. ओके, तो इसका मतलब हमारा जो इंडियन लाइफस्टाइल is known to have some kind of mutational risk with respect to gut microbiota or diet patterns or whatever. Okay, next slide please. And then we ask, ठीक है हम data generate करते आ रहे हैं, हम big data हम खेला रहे हैं, can we try to come up with the predictive analytics? Can we really compare this with future data sets? यदि 10 years down the line, यदि कोई बंदा अगर ग्रोन करके या पेन के रूप में यदि आता है, और हमारा इंडियंस में ऑलरेडी इनहेरेंट स्टिग्मा है कि यदि हमारे पास कोई पेन आता है जेनिटल्स में, इसमें डॉक्टर के पास आते नहीं है, हम आखिर हम वाइफ को भी हमने बताते है कि नहीं. So we might be deprived of that inherent stigma. So what we thought was, can we really come up with a kit just like a pregnancy kit? Okay, urine drop करो और आप प्रोस्टेट कैंसर विक्टिम है कि नहीं, हम एक बायोमार्कर एक हम आइडेंटिफाई कर सकते हैं. That is what we thought. So this is what predictive analytics is. Next slide please. So in order to do that, जब हम ये कर रहे थे, interestingly SARS-CoV-2 में भी ये particular prostate cancer के long non-coding RNA mutations भी थे actually. Other way around, this work is still ongoing, but we wanted to tell you one particular take-home message here is कि यदि कोई प्रोस्टेट कैंसर का विक्टिम है, बंदा है, it has been proved that he is more likely to survive from COVID than a person who doesn't have a prostate cancer. That's because वो बंदा इतना resistant बन जाता है non-coding RNAs रहने की वजह से. This is one of the reasons why these non-coding RNA repositories are playing a very, very important role. Okay. So next slide, please. So maybe in the near future, 10 years down the line, we have started a consortium. So whole exome sequencing data ले लो, regulatory data ले लो, long non-coding RNA data ले लो. Okay. And then you identify the variants and come up with a machine learning approach. हालांकि मैं आपको फर्स्ट स्लाइड में जो मैं आपको बताया था, प्रिडिक्टिव बेस्ड अप्रोच, प्रिडिक्टिव एनालिटिक्स आप निकाल सकते हो. ठीक है. एंड देन मे बी ये फीचर है, मतलब इन पर्टिकुलर बंदा में ये सिम्टम्स हैं और ये जीन म्यूटेशन है और ये है, यदि आप सिंपल ब्लड टेस्ट से यदि आप आइडेंटिफाई कर सकते हो या यूरिन टेस्ट से आइडेंटिफाई कर सकते हो, देन यू कैन रिड्यूस द कॉस्ट ऑन ए लार्ज स्केल. Okay, so that is what we are trying to achieve with this particular project. Next slide please. So the conclusions for this part 2 of the case study is, we have identified mutations largely from prostate cancer victims who are vegetarians, largely associated with diet as well, largely associated with obesity and vitamin D and mitochondrial dysfunction as well. Okay, and finally we have developed a conceptual framework. मतलब हम एक hypothesize ये हम कर रहे हैं कि prostate cancer is not necessarily related to the eating habits of non-vegetarianism, but it could also be associated with diet, lifestyle, and whatever obesity, motapa, whatever you want to say. Okay. Next slide please. The third what do you call part 3 slide. Next slide. Yeah. So what we have done in this particular part 3 is, next slide please. Yeah, Pankaj, I'm sorry, thank you so much. I think हम आपको बहुत कष्ट दे रहे हैं. So this is a very paper, this paper came up a couple of months ago. हम SARS-CoV-2 में भी हम करते आ रहे हैं. This is a very something close to my heart, a very important systems approach. What we have done was, usually आपको पता होगा कि SARS-CoV-2 में 2 रिसेप्टर्स को रिसीव करता है ह्यूमन. ओके, मतलब SARS-CoV-2 इन्फेक्ट करता है ह्यूमन में. AS2 और TMPRSS2 2 जीन्स हैं जो वेलकम करता है. ठीक है, मतलब सेल के भीतर बैठ के आप आइए, SARS-CoV-2 को रिसीव करता है. Now what happens is, when SARS-CoV-2 is being received, there are other genes, other non-coding RNAs that keep watching. गौर से देखता रहता है कि इन SARS-CoV-2 कैसे रिसीव कर रहा है और कैसे सेल को इन्फेक्ट कर रहा है करके देखता रहता है. Now what we identified was, there are some non-coding RNAs, okay, that transgress, okay, that are being transgressed. मतलब SARS-CoV-2 क्या है, long non-coding RNAs को transgress कर देता है. मतलब ये AS2 को भी अपना लेता है, and at the same time some long non-coding RNAs present in the cell को भी अपना लेता है. Okay, it owns it. Okay, it takes the entire long non-coding RNA basis into its own. Okay, and this is what it really happens. मतलब SARS-CoV-2 इतना intelligent हो रहा है, आप देखिए, and this is one of the reasons why Omicron variants are coming up. Okay, next slide please. And we have identified some unique epitope signals which are seen in capital letters in bold. Okay, there's all the conserved sequences and epitopes which play a very important role in those ORFs. Next slide please. तो मैं आपको ट्रांसलेशन जब आपको बता रहा था, NSP15 okay, non-structural protein 15 of SARS-CoV-2, वो ट्रांसक्रिप्ट कर देता है लॉन्ग नॉन कोडिंग आरएनए, मतलब एग्जैक्ट जो बेसिस हैं ऑफ लॉन्ग नॉन कोडिंग आरएनए कॉपी कर लेता है, and then it reinfects the entire cell, and as a result pura mitochondria pura they die, and if mitochondria dies, they are the powerhouse of the cells, okay, then the asphyxiation takes place, okay, and as a result the cell leads to death, and as a result the person also dies. So this transcription hypothesis ये भी हम systems genomic integration में हम कर चुके हैं. The next slide please. Yeah. So इसमें ये मेरा पोस्ट डॉक प्रोजेक्ट था, 2012 की बात था, 13 की बात था. जेनेटिक्स में सिस्टम्स जीनोमिक्स अप्रोच में एक वेरी इंपॉर्टेंट थिंग, आपको स्निप्स आपको प्रायोरिटाइज करना है, ओके. हालांकि आपको कई बार ऐसा होता है कि मेनी अ टाइम यू फाइंड सर्टेन जेनिक लोकेशंस, ओके, इन दोस जेनिक लोकेशंस यू डोंट नो, अगर 2000 बेसिस रीजन में, एक स्मॉल लोसाई में कम से कम 1000 बेसिस आपको म्यूटेट होने का संभावना है. That is what we call it as linkage disequilibrium in genetics, okay. So अगर आप 1000 bases of 2000 bases, मतलब हर 2 बेस में आपको म्यूटेशन यदि मिल रहा हो, आपको कैसे पता कि वो particular mutation is detrimental to the disease or which mutation is really causing that particular disease condition, okay. And that too if you have a pleiotropic environment. Pleiotropy is ऐसे जरूरी नहीं है कि एक जीन एक ही डिसीज करे, कॉज करे. You might want to know that one gene might also cause several other diseases. Like BRCA1 doesn't necessarily cause prostate cancer. It also causes breast cancer. It is also causing, xeroderma pigmentosum or all other DNA repair diseases. Okay, so that is pleiotropy. So which means not necessarily one gene is known only for causing one disease, but it is elevating, different diseases. तो ये प्रॉब्लम था हमारे पास, मतलब एक ही स्मॉल जगह में इतने सारे बेसिस पड़ा हुआ था, एंड यू डोंट नो व्हाट काइंड ऑफ म्यूटेशंस आर डिट्रीमेंटल टू योर डिसीज कंडीशन. तो हम क्या किया, म्यूटेशन स्ट्रेटेजी हम एक आइडेंटिफाई किए. नेक्स्ट स्लाइड प्लीज. So we have looked into those genome browsers, identified those mutations, which mutations are present in the transcription factor binding sites, which mutations are present in the open chromatin elements, and all those kind of regions. Next slide please. तो 3 different approaches लिए annotation approaches में. पहले functionally annotate किए, उसके बाद constrained elements निकाले, उसके बाद regulatory regions में कोई SNPs है क्या वो भी निकाले, okay. And then we identified whether or not these mutations are, detrimental to the deceased condition, okay. And then these conditions play a very very important role, because आपको एक important चीज निकालना होगा कि ठीक है, एक different annotation type से different mutations निकाले, but क्या पता ऐसा होगा कि आपके पास कुछ mutations तीनों annotations सिस्टम में शामिल होने का चांस है. मतलब एक म्यूटेशन आपको फंक्शनली एनोटेटेड स्पेक्ट्रम में मिलेगा, और सेम एनोटेशन से कंस्ट्रेंड एलिमेंट में भी मिलेगा, सेम एनोटेशन रेगुलेटरी रीजन में भी मिलने का चांस है. तो कैन वी से कि that particular SNP can be detrimental to the disease? Can we say that particular SNP might be pathogenic in nature? That is what we thought of doing that, okay. Next slide please. So this is what this circle shows. देखिए ऑरेंज वाला, अगर आप गौर से देखेंगे, ऑरेंज वाला सर्कल्स सारे पाई में हैं, okay, which we call them as disease SNP, because एक पाई एक annotation strategy को कहलाता है, okay. So if this particular SNP is seen in the functional annotation strategy, the same SNP is also present in, say, in the yellow one, which is, say, constrained element, and if the other SNP is also present in the regulatory regions in the green color, then we can say that yes, वो SNP disease cause करने का chance है. That is what we have developed an annotation strategy, and that is a causal SNP or a deceased SNP. Next slide please. So the take home message for this particular 4th case study is SNPs or variants identify करना इजी बात तो नहीं है, बट आप एनोटेशन स्ट्रेटजी और एनोटेशन स्ट्रेटजी के रूप से आप प्रायोरिटाइज आप कर सकते हो. That prioritization plays a very important role. So we have developed our own prioritization strategy using 3-point classification approach. Okay, and this is what we have done. We have used different functional annotation, जितने भी जो SNPs enhanced elements में हैं, methylation और regulatory approaches में हैं, और किन SNPs pathways में होने का chance है, हम सारे निकाले हैं. एंड वही स्निप्स हम अपना लिए जो डिसीज कंडीशंस को और कॉजल स्निप के रूप से वो डिफाइंड है. ओके, मतलब if one SNP is associated with all the 3 different annotation strategies, we call that particular SNP as a detrimental SNP or a disease SNP. Okay, next slide please. So the final take home here is prioritization in systems genomic plays a very important role. आपको सिर्फ सीक्वेंसिंग करना या जीवा स्ट्रेटेजी से आपको म्यूटेशंस आइडेंटिफाई करना इज नॉट एन इम्पोर्टेंट. ओके, वो इजी है, बट आपको प्रायोरिटाइज करना, मतलब किन म्यूटेशंस आपको डिसीज फैलने का प्रायोरिटाइज कर रहा है, वो आपको निकालना. सो ऐसे कई बार ऐसा होता है कि ऑल कॉजल वेरिएंट्स में भी एक कैंडिडेट बोनाफाइड SNP. Okay, but not all bona fide SNPs can be considered as causal variants. आपको validate करना होगा. Validation किस रूप से करना होगा? ऐसे annotation strategy से आपको निकालना होगा. whole exome sequencing Sanger validation करना होगा. Okay, कुछ experimentation mutational PCR करना होगा. Okay, mutational spectrum, all these things that you need to do at Goodwill. Okay, next slide please. So I think I'm coming at the end. So before I close in, I'll just try to tell some other things. I'm a part of this topic called Integrated Systems Genomic Approaches. If you have any article, please send us at prash@biocruise.org. You can go through that particular topic as well. You can take the screenshot. Next slide, please. एक और Systems Genomic Approaches for Understanding Multi-Omic Data, MDPI में भी है, वो भी आप open है next 30th August 2022 तक. So if you have some articles, you are most welcome to send me. Next slide please. What I'll take another 5 minutes. I know हम सारे सब लोग क्या है कोविड के तौर से बहुत alarming है. We are really sitting at home. हम faculty है, but we are still facing some kind of problems. But the most important thing here is the students are really facing a hell lot of problems. If you can see here, I'm just curious. I just want this to be more interactive because you're all faculty and you'll be teaching your students as well. What do you see from this slide? Can anyone answer quickly? Yeah. I'll come back with the questions, please. Yeah. Can someone tell me? You can unmute your microphone, maybe, or chat. I don't know. Yeah. Yeah. Anyone, please? What do you see in this slide? Anyone? Yeah. Okay. Researchers containing COVID. Okay. Anything else? Anyone else? Yeah.
We are carrying the burden of COVID.
Corona being carried. Okay. Yes. Next.
Sir, are you able to hear me?
Yeah. Sorry. Yeah. So you can see, sare gentlemen are going forward, leaving a woman behind, which I feel is a bit unfair, isn't it? Okay. और वो बेचारा एक बंदा है, एक छोटा वायरस लेके जा रहा है. He is not even looking at the back, how a woman is not taking that load. Okay. We हम तो बहुत गप्पे मारते रहते हैं घर पे बैठ के कि that we are sitting at home, probably helping everyone here. But at the back of this particular mind here is we have, the mitochondrial progenitor in the form of a woman who is carrying this particular. I firmly believe that if the women takes lesser load from this particular stress, we can move, pretty happily, towards the future. And this is one of the reasons why many scientists are probably not that productive. Okay, this was shown in a PNAS article by Malish et al. In 2020 when the COVID, came into picture. So gender imbalance is taking, a proactive role, unfortunately, post-COVID times. Okay, next slide, please. So to overcome this, we can do some kind of, strategies. We can communicate well. We can maintain some moral discipline, moral policing. We can bring some kind of, social norms. Get down, some kind of social inequality. Okay, we can keep away the prejudice. We can keep away the stress and cope up factors, pretty well, and all those kind of things. Next slide, please. The best thing is, you can remove the trash in your mind and also in your mobile phones and also in your laptop as well. Okay, because घर पे बैठ के we were not polluting the roads, but we were polluting our, several, messages that is putting in our mobile phones. Okay, that is also playing a very important havoc, where अगर unused apps अगर आप नहीं करेंगे तो ये environmental impact के रूप से भी, फैलने का chance है. Okay, this is not just in theory but also in practice as well. Next slide, please. So this is where, we have piggyback between two important syndromes called imposter syndrome and Dunning-Kruger syndrome. And imposter syndrome, हम क्या है डाउट करते रहते हैं हमारे कैपेबिलिटीज के ऊपर. ओके, दैट इज व्हाट वी कॉल इट एज इम्पोस्टर सिंड्रोम. अरे घर पे बैठ के क्या होगा, ये एक्सपेरिमेंट क्या होगा, हमारा जॉब का स्टेटस क्या होगा, ओके, हमारा पब्लिकेशन का क्या होगा. दैट वाज व्हाट we were piggybacking in 2020 at some point. Okay, but more than 2020 April, when the COVID was came into picture, we also had something called Dunning-Kruger syndrome, because we were very overconfident. ठीक है, summer आते समय, COVID चला जाएगा. Pankaj, I cannot see the slides. Can you see it? I'm sorry. Yeah. Now I could see. Yeah. So COVID चला जाएगा, summer आते ही virus चला जाएगा. But you'll be surprised to know that COVID has shown more effect only during summers. अगर आप देखेंगे तो पूरा March to May of 2020 and 2021 में ही ज़्यादा infections फैला हुआ था. This is alarming. But maybe by 2021 December में है, okay. But COVID को आप consider it as a a blessing in disguise. COVID है इसलिए we are facing some kind of acute challenges and how to overcome that. यदि COVID नहीं था, we would not have probably overcome such kind of challenges. So this is my important take-home message. The next slide, please. So we need 3 C's: collaboration, convergence, and consistency. These are the 3 important things where we can overcome the human activity on the crisis. The next slide, please. So I'm also the founder of BioClues, India's largest bioinformatics society, which we have been doing over the last 18 years. Tremendously working towards development of mentor-mentee relationships. We have organized a lot of faculty development programs, a lot of events, a lot of mentor-mentee relationships, published several papers, and we have a prestigious event called Indian Conference on Bioinformatics, which was held in CSIR NEAST in last month. Okay. This Index 2022 we are organizing in Guntur and Hyderabad. Okay, and we are very pleased to announce that Sir Rich Roberts, Nobel laureate for alternative splicing, has accepted to give the keynote. So you're most welcome to join our mailing list, bioclues.org. Okay, and then I'll be happy to keep you updated on this. Okay, next slide, please. So this is my final slide. I look forward for some Q&As. I thank Pankaj for this invitation, for his kind invitation and gracious time today, and also the technicians who helped me get through this. all these works, the publications, the wonderful publications over the last several years, is not possible, without all these wonderful people and all my research group members, all my 7 PhD fellows and 2 postdocs who work with me and who are working with me as well. And of course, I thank our Vice Chancellor and our Chancellor, Amma Amritanandmayi, and also our Dean, Dr. Bipin Nair, Amrita School of Biotechnology. So I look forward for any questions. If you have any questions, I'll be happy to answer.
Thank you so much, Prash. Wonderful interactive session.
Sorry, I'm not able to see the chat somewhere. I don't know. Yeah, it's here, chat.
Is it,
yeah. So my chat box is on. So if you have any questions, I'll be happy to answer.
There are a few questions that have been already posted by Rajesh Anand, and one of the question, maybe let's go through them. Is Rajesh already in the panelists?
Yeah, I'm sorry, I cannot hear. 1 minute, please. Yeah, 1 minute. Yeah, yeah, please. Yeah.
So one question that Rajesh has put is, I'm interested in differential analysis of genes and proteins, and which direction is better and cost effective, starting from proteins to transcriptome or transcriptome to protein?
Yeah, I would always say transcriptome to proteins, because it's always interchangeable. If you have some differential express genes that you identified from RNA-seq or whole transcriptome shotgun sequencing, many a time those differential express genes could also be submitted with differential express proteins as well. Okay, which means यदि आपके पास RNA-seq से अगर आप differential express genes अगर आप ले रहे हो, then proteomic तौर पे अगर आप MALDI-TOF या simplest top-down quantification अगर आप करते हो, या nano-LC करते हो, या mass spectrometry से अगर आप identify करते हो, then whatever differential express genes from your samples were differentially expressed, you can definitely say that those genes are also translatable to differentially expressed proteins as well. Okay, so I think from transcriptomics to proteomics is always a good idea. But you can do smaller number of transcripts, transcriptomic study, and go for bigger number of proteomic based study. That can be an ideal one if you don't have budget. Yeah, I hope that that helps, Rajesh. Yeah.
Thanks, Parash. There is next question for you again from Rajesh. If we find mutations from our sample in clinical samples, how do we validate their clinical significance?
Yeah, so as I told you, the validation step is based on the depth of the coverage. So you should have a larger depth for every sequence, at least a minimum of 5 or 10 bases, 10 depth should be there. And that validation is usually done using something called Sanger validation chemistry. And the most important thing is not only the depth and the Sanger validation step, but you will also talk about the minor allele frequencies. Okay, which probably I missed telling you that. So in a minor allele frequencies, usually अगर Indian human subpopulation अगर देखा जाए, तो 5% of variation, which means minor allele frequency of less than 0.05 is a kind of an ideal one. Okay, so 5% of that minor allele frequency is a kind of a stepping stone, for success. Okay, so अगर आप rare disease कर रहे हो, तो 0.01 strategy आप employ कर सकते हो, which is 1% of your minor allele frequencies. Okay, that is where you can start employing your validation, and you can validate all the SNPs that you want, but usually 5% of the SNPs that you want to identify and consider them as pathogenic can be validated. Okay, Baki, it's only for your publication front, but if you want to develop a diagnostic panel, you have to validate all the mutations that are known to be pathogenic in nature. I hope that answers your question, Rajesh.
Yeah, sure. Next question again from Rajesh. So how do we use RNA-seq data to search for rare variants?
For real variants?
Rare variants.
For rare variants, I don't think RNA-seq could be an ideal choice for rare variants, because RNA-seq यदि आप कर रहे हो तो उससे अच्छा whole genome sequencing आप कर लेते हैं. RNA-seq is largely used for probably for identifying the transcripts or gene expression profiles. Okay, but variants अगर आपको निकालना हो तो then that's a different strategy. Okay, you can still do that, but I wouldn't advise that. The best thing is to go for whole exome sequencing. अगर आपके पास budget है, rather RNA-seq, I would ask you to go for whole genome sequencing for at least small number of samples, or maybe the targeted genome sequencing for larger number of samples. Okay, I hope that answers your question. Yeah.
Sure, Pras. Is there any other question? I don't see any other question in the chat box, at least. Okay, so I think you can, if participants want, they can reach out to Dr. Prash directly for further conversation. Before, so let me thank again Prash, and before you leave, I would like to take one screenshot, so just as a proof that we are all here. If you don't mind to turn on your video just once.
Yeah.
Thanks. I will just...
I don't know why your voice is a bit feeble, Pankaj. I'm really sorry. I got a call. Maybe उसके वजह से कुछ ये change हो गया था. 1 minute. Yeah. 1 minute.
I'm sorry. Is it audible now? Better? You are not visible properly, Prash. Can you try to shift your mobile once?
Yes. Yeah. Can you see me, Pankaj? Yeah.
You disappeared. I cannot see you.
Yeah.
Yeah. Now it's better. Thanks. Just hold on. There we go. Yeah. Thanks. Thanks, Prash. Thank you. Am I audible, Prash? Hello, Prash. Yes. Thank you so much. I hope I am audible to you.
Good morning, Pankaj sir. Good morning. Please let me know my voice is audible.
Yes, you are audible. So let me first thank Prash again. I hope he can hear me.
Sorry, Pankaj, your voice is not audible to me. I don't know why. All of a sudden. Yeah. Now, can you please speak again? Yeah.
Yes, I was saying thanks a lot, Prash. I hope it is audible now.
Yeah, it's audible, but I don't know why it has drastically come down. It's okay. I'm just I switched on my camera so you can see my face, I guess. Yeah.
Okay,
so is the picture taken, Pankaj? Can I put off?
Yes, yeah. yes.
Oh, thank you. Oh, I don't know this. Yeah, thank you.
Thank you, and we will move to now the next speaker, that is Mr. Deepak Yadav. So Rajveer, please take over now.
Yes, sir. So, good morning, everyone. It's a great pleasure for me to introduce our next speaker for today's talk, Mr. Arpit Deepak Yadav. So, Mr. Arpit completed his BE in Electronics and Telecommunication from JDIT, Yavatmal, and then MTech in VLSI Design from GHRC, Nagpur. Mr. Arpit did a PGP in Artificial Intelligence and Machine Learning from University of Texas, USA. So currently, Mr. Arpit is pursuing PhD in Machine Learning from SVVV Indore. He has filed 4 patents, 3 Indian and 1 Australian, in artificial intelligence. He was awarded a VD Good Inspirational Scientist Award in 7th International Scientist Awards on Engineering, Science and Medicine, and also Excellence in Research Award. He is working as a senior data scientist at International School of Engineering, Bangalore. He has done 70 plus certifications and also a corporate trainer in the domain of data science, machine learning, deep learning, and artificial intelligence. And he is also an active researcher in artificial intelligence and machine learning. So, without any further delay, please join me in welcoming Mr. Arpit Deepak Yadav to address the session on breast cancer prediction. Over to you, sir.
Thanks, Rajveer, for your kind introduction about me. Before starting the session, please let me know my screen is visible and my voice is clearly audible or not.
Yes, sir, you are audible and your screen is visible.
Okay. So I'm starting my session. Thank you very much. Okay. I welcome all of you. Very good morning to everyone. I welcome to you in the AICT sponsored short-term course on Multi-Omics Data Science, which is organized by the Department of Bioscience and Bioengineering, Indian Institute of Technology, Jodhpur. I would like to thank all the organizers, those who have organized such a wonderful event, so that participant can learn the upcoming technology and they can upgrade their skills for the future benefit. Before starting the session, I would like to say something to all of you. Always remember one thing: whenever you are planning to learn any new technology in the market for upskilling yourself, so directly before jumping into that technology, first ask the 3 questions to yourself. What are those 3 questions? The very first question is, you need to ask that, what is this technology about, what I want to learn, and why it is so much famous in the market. Second question you want to ask is, why all the multinational companies are using this technology, and what are its impact on our society and its application. And the third important question is, what are the skills required to learn this technology, and after upgrading that skills, what are the opportunities for me in the market in terms of job, in terms of research, or either in terms of startup. Today's my topic of the discussion will be cancer prediction, breast cancer prediction using machine learning algorithms, that is with the help of artificial intelligence technology. And I will be answering all these questions with a very quality guidance. So be with me during the session. My name is Arpit Yadav and I'm working as a senior data scientist and a researcher in AI ML and the CTO at AIBI Bangalore. So let's start the session. As Rajveer has already given my introduction, that I have completed my bachelor's in ETC, Electronics and Telecommunication Engineering, and master's in VLSI Design. I have completed my post-graduation program in AI ML technology from Texas University, USA. And currently I'm pursuing my PhD in machine learning. I'm having the total 12 years of experience, out of which parallelly along with my main job, I'm having the 8 years of experience in the skill development training across the India. And I'm very passionate to give the skill development training in the recent technologies. I'm an international speaker and the mentor in the field of artificial intelligence and machine learning technology. Till date, I have filed 4 patents. I have done so many certifications. And as I'm currently working with the 2 organizations with a full time, where I'm working as a senior data scientist at INSOF Bangalore and chief technological officer at AIBI Bangalore. Parallelly, I'm associated as a mentor with multiple companies and the universities across the India, where I'm working as a mentor in AI/ML technology at BITS Pilani. I'm working as a mentor in AI/ML program at IIT Guwahati, whereas I'm also responsible for designing the curriculum of data science, machine learning, deep learning, and AI technology across the world. These are the various universities. And after that, whatever the small contribution I have been done in the field of AI, I have been rewarded with the 4 awards. And these awards motivates me a lot to do something better for the society. So these are those awards which motivates me. Everyone needs a recognition and motivation to do something better. So these awards motivates me, and the passion which I am delivering the session across the India motivates me to impart the knowledge to the upcoming generation. So let's start today's main topic of the discussion. Our today's main topic of the discussion is the breast cancer prediction using the machine learning and artificial intelligence technology. So before I want to tell one thing, today's my session is divided into 2 parts. The very first half, I will be delivering what is the meaning of data science, machine learning, deep learning, and artificial intelligence technology. Because I believe that many participants might be not knowing about this technology. And then after that, I will be giving you the process that how we are applying the data science and machine learning technology into the genomics domain, and how we are achieving the different kind of results, how we are achieving the today's topic result as well, prediction of cancer. So let's start the process. So please remember what I told you in the very first slide. Whenever we are learning any new technology, so today's technology that we are talking about is data science and machine learning. So when we are learning data science and machine learning, so we need to ask the 3 questions related to this, that is this: what is data science, machine learning, deep learning, and AI? Why it is so much famous in the market? why all the multinational companies, small scale companies are using this technology, how it is impacting our society and day-to-day life, and what are the skill set required to learn this technology, so that after upgrading the skill set, I should apply in my own particular domain for research purpose, for job purpose, for startup purpose. So I will be guiding all this in a very easy way. So please concentrate on this diagram. It is extremely important to know the following equation in the slide. Today's new normal technology is the AI technology. It is a new normal world right now. Everyone and everywhere in the industry, AI world is going on. But why? What question is why? So the answer is very simple. That data science, machine learning, deep learning, and AI technology, when you are adding with any X, I will let you know what is the meaning of X. Your answer is always profit, growth, or a business. That means your result will be profit and growth. So here X means any industry, any domain. Whenever you are applying data science, machine learning, deep learning, and AI technology with any X, X means now put X as an automobile industry. Data science and automobile is equal to profit. Put healthcare industry. Data science, machine learning plus healthcare industry is equal to profit. So in place of X, you can put any domain in the market. Either it is a banking, healthcare, agricultural, space technology, education industry, manufacturing, transportation, anything. The slogan in the market is right now, not a single industry or a domain left in the market, those who are not using the data science and machine learning for their growth. Hence, it is the right time for all of us also, all the participants, those who are listening this, you are having the domain knowledge, might be your particular domain, you are an experienced person, might be. So you are having the experience in some particular field. In that field also, data science is applicable. So you can add the data science and machine learning, do the research, make some product, and have a profit in that. So hence this technology is called as a new normal technology, which is applicable everywhere. And now in our today's session, I will be introducing you the world of data science, machine learning, deep learning, and AI in a very layman term, so that anyone can understand this. And then after this, you can able to apply this technology for your future studies. So in our entire session, I am going to let you know what is this data science and machine learning technology about, who can learn this, why it is so much famous from technical point of view in the today's world, how we inside a company are doing any project of data science and machine learning, what tools and technology and softwares we are using for doing the projects in the data science and machine learning and AI, what are the skill set required, and after upgrading the skill set, what are the opportunities in the market, what are the challenges, and everything I'm going to discuss in a very layman term so that anyone can understand this. So whenever I'm giving my session across the world, so first doubt of the participant is coming that who can learn data science? I'm from the XYZ field, and whether it is possible for me to learn the data science technology or not. Participants might be an experienced participant. Those who are having the experience, they are asking that I am from XYZ field, I am having the N years of experience. Students are asking the doubt, I am from the different background, commerce, arts, science, in the engineering, different branches. So whether it is possible for me to learn or not. Or if it is possible, then what is the exact right time to learn this technology? So for this purpose, I am having something very motivation things to show you. I am showing in my every session that small students, school going kids are learning this technology. Government of India has given notification to all over India to teach this technology. So for the motivation purpose, he is Mr. Siddharth Pillai, 7th standard student in Hyderabad, working as a data scientist in the company, 7th standard student. Okay. And he is Mr. Tanmay Bakshi. He is a IBM researcher at the age of 16 year, 16 year. He is an international personality giving the TEDx talks as well. Just a 16-year-old boy. Then after that, he is Mr. Sarang Sumesh, 8-year-old boy, making the artificial intelligence robot. And his episode has been telecast in the History News channel. He is a TEDx speaker, youngest TEDx speaker. And finally, I got shocked when I came to know about him. He is Mr. Arham Om Talsania, who has cracked the Microsoft Python examination, and his name has been recorded in Guinness World of Record. Okay, why? Because he is the youngest in the world who has cracked the examination. Why I'm showing this? Because the thing is that those who are having the fear that whether it is possible for me at this age to learn this technology or not. So obviously it is possible. You can learn this technology with a very easiness. And when to learn this? Because we are already late. Everywhere right now, data science, machine learning, deep learning, and AI is coming for their for any kind of industry growth. So the answer is yes, we people can learn this technology, provided you should have the learning attitude, time and patience, and obviously you can learn that. I'm going to provide you the right guidance today with a proper roadmap to apply data science in any kind of domain. So after knowing this, guys, the main question arises that why should we learn data science and why should we learn the AI for any kind of growth purpose? So let's go ahead and go deeper into this. Actually, every multinational companies in world or every research institute in the world are gathering the data for the different purposes. Every company is having the data in their database. Now what the company is thinking, I want to utilize this data for some business purpose and for some growth purpose. So how they are utilizing the data? They are using that data for some different projects. And on the basis of that project, they are solving the problem of demand and supply of the society, and they are doing the business. Because of this reason, every company has declared that data is the new fuel of 21st century, because on the basis of data, they are doing the business. And hence, by knowing the impact of data to the nearby future, all the multinational companies has declared that data science is a very attractive career because it is changing our world day to day. How it is changing, I will let you know. And it is the future. Hence, there is a necessity that every professional and the student should at least learn the data science and apply in their own domain for the profit purpose. So after knowing this, the main thing is that now in your mind, what is the exact meaning of data science now? I know that there is a need of data science because everywhere data is there and we want to make a business from that. But what is the exact meaning of data science? In the internet, there are thousands of definitions are there, a flood of information. But the exact and a good and easy meaning of data science, I'm going to tell you. The meaning of data science is very, very simple. That science of data to make a business. What is data science? Science of data to make a business. That is making data work for you. Very simple. In your mind, this question might be arise that how we are going to make a business and what kind of science of data you are talking about. We want to make a business and what kind of science of data you are talking about. So let's check it. Actually, as I told you, every company and research institutes are collecting data, and that data is called as a raw data. Now this data science technology, what it is doing, they are applying some methodology on raw data that is called as science, and they are extracting the meaningful information from the raw data. So converting the raw data into meaningful information is a data science world. Okay, that is making data work for you people. And actually, I'm having the habit of teaching the technology from the word itself. Now, data science, having the two word, data and science. Break the data and science into two parts. Data is separate and science is separate. What is data according to company? Data is divided into the three category actually: structured data, unstructured data, and semi-structured data. And science. So according to the world, structured data means what? For example, we are having the Excel sheet and we have collected some data. In that Excel sheet, all the data is properly filled. No data is missing. Everything is properly filled. Such kind of data is called as a structured data. Everything is neat and clean. Then what do you mean by unstructured data? Unstructured data means data available in the form of images, video, audio, and text. That is unstructured data. And according to one analysis in the world, it has been said that out of 100% data which is available in the world right now, 80% to 85% data is unstructured data. So we are having the availability of such kind of data when we are collecting the data for different purposes. So now the challenge is that now we have collected the data and keep in the database. Now what we are doing as a data science professional, we are applying science to that raw data which is collected. And what is that science? So science is the combination of 4 things. What is that? The combination of one programming language, one mathematical aspect, that is programming language, second is mathematics, third is algorithm knowledge, machine learning and deep learning, and the fourth is visualization knowledge. These 4 things are called as the science, and this 4 combination, when we are applying on the raw data is going to give you meaningful information. And from that meaningful information, companies are making the business by taking certain decisions. This is the simplest form of data science to understand. That means we are having raw data, apply science to that raw data, collect meaningful information or either research knowledge and do a business. That is the simplest thing. This is the data science world. Okay. And after this, the next question should arise in your mind that from where the data is coming, from where this data is coming, from where company is bringing the raw data. So for that purpose, we all people are responsible. We all people are responsible. How? Every day we people are typing the messages in WhatsApp, Facebook, Instagram. We are uploading our photos. We are uploading the videos. We are doing online shopping. We are doing online cab booking and many things. It indicates that intentionally and unintentionally, we people are generating data which is collected by the companies. And every day, either online or offline, we people are generating the data. We all individual are doing that. And because of this reason, companies are collecting the huge volume of data. And from this huge volume of data, from our own data only, companies are making the billion-dollar monies and businesses. And we are happily giving our free data to them. In the Facebook, I'm having the Facebook account. Every day I'm uploading something, some news and anything. So my data free of cost used by the Facebook company, and they are growing. Okay. And it is not wrong to say that I am also having the contribution to make Mark Zuckerberg one of the richest person in the world. I am having the Facebook account and free of cost my data is with them. So you can imagine that intensity of your data from your own data free of cost, other person are growing. And just because of this reason, nowadays, today's world is called as a data driven world. Why data driven world? Because on the basis of data, everyone is growing, everyone. And because of data, everyone is growing, everyone. And because of this reason, it is said that data is your new oil, information and asset. We are living in the Industrial Revolution 4.0, and it is the revolution and era where any technology which is data dependent are going to boom the market. Artificial intelligence, machine learning, deep learning, data science, data dependent. IoT, blockchain, Salesforce, data dependent. All are booming in the market because all are having the dependency on data. After knowing this, how this data science is impacting our day-to-day life. Whenever we are doing our education, so these are the 3 main streams we are deciding to the education. Either science, either commerce, or either arts. You choose any one of them. The student can choose any one of them. Those who are in the school right now and in future, they are opting this. They are going to get data science in their domain, compulsory. Because everywhere industries are in the commerce, industries are in the arts, industries are in the science domain, and everywhere data science is applicable. And as I told you, if someone is taking the admission in engineering, so data science and machine learning is applicable to every engineering branches. There is no need to take any tension also. Either whatever education background might be you are having, you apply data science in your education background also to form some product with a combination of data science and your education, and you can have a growth in future. You can have a growth in future. Plus, that is about education. Let's talk about industry. Every industry here is called as a domain. Data science and machine learning is applicable in every domain. When we talk about domain, it is an industry. You apply in the healthcare, aviation, finance, agricultural, and so on. Not a single domain left in the market. Those who are not using the data science for their growth, they are applying for that. We are collecting the data. Even though today's our topic is on genomics, all the genomics industries are also using the data science and AI to know what should be the pattern in future, prediction purpose. Okay, and we are going to check this today, how we are going to predict the cancerous cells with the help of data science and machine learning technology. Okay, first half just I am explaining you that what is the meaning of this, then practical I am going to explain you. After knowing this, that data science and machine learning is impacting the world in every domain, and that's what hence you can observe in your surrounding that in every education background also data science and machine learning branches are opening. Every industries is asking their colleagues and their employee to learn this technology and upgrade that. And that's the important thing also according to me, because we are getting so many projects in the industry from the different domains. And after this, up to this slide, what we came to know that data science meaning is that extracting the meaningful information from data, raw data. And from that meaningful information, companies are making the business. But how to extract meaningful information? That is the skills. And for that skill set only, companies are hiring the data science professional. You should have that skill set to extract the meaningful information. And that skill set, as I told you, knowledge of one programming language, that is Python. I'm going to tell you that. Then mathematics, not complete mathematics, school level mathematics. Then algorithm knowledge and data visualization. Then you can win the race. You can apply on any domain and you can go ahead with that. And why this technology is so much famous in the market? After my talk, if you will search in Google that what is machine learning, so you will came to know that machine learning is already invented in the year of 1959. Today is 2021. Why this technology is famous from last 10 years? Why not it was famous in the year of 1959 when it is invented? Because of the following reason. These are the 4 following reasons why this technology, AI, data science and machine learning is famous today and from the last 10 years. And these are the reasons. As I told you, today on a single click, data is generated. So easily availability of data in our today's environment. Second, today we are having the powerful computer systems. Powerful computer system. When I'm talking about powerful computer system in 2 terms, powerful memory capacity of computer and powerful processor capacity of the computer. Then today we are having the powerful algorithm. Algorithm means the world of machine learning and deep learning. Here algorithms are there. And the 4th is open source software tool. This 4 combination today is available very easily, and because of this reason, data science and machine learning booming the market. Before 20 years, we are not having the powerful computer which can process the huge volume of data. But today we are having that. Even though our laptop is having the 1 TB of hard disk nowadays. So you can do a projects in this, and you can open your startup also, because all the open source software tool is available free of cost. So because of this reason, this technology is very famous. And after that, this is the roadmap to learn the AI technology for all the people, those who are listening this and planning to learn AI. So you want to follow this roadmap. And this is the step by sequential roadmap. You cannot go directly and jump into the other. To learn the AI technology, you want to first learn one programming language that is called as Python. Why Python? There are more than 10 programming language available in the world of AI. Why I am only talking about Python? Because Python is one of the most easy and dominant language in the world of AI. And so many powerful packages available, easy to learn. Anyone can learn Python. School kids are learning Python. Then after learning Python, we are going to learn data science. Data science means the analysis world. We are having the data. We have to analyze that data with the help of Python language. And the meaning of data science to clean the data. Raw data is there. We have to clean that raw data. And the output of data science is going to machine learning. Then here machine learning is the world of algorithms. We are going to apply the algorithm to the clean data. So that algorithm will learn from the data and output is a model. Then certain drawbacks of machine learning, which is overcome by deep learning. Deep learning is the world of neural network. And here also we are doing the same, applying the algorithms. And then after learning deep learning, AI, you will go and learn the AI. So if you will follow this stepwise roadmap, at every roadmap, jobs are available in the market, every roadmap. If you will learn Python, forget about the topmost floor, you will get a job of Python developer. If you will learn the 2 stages, Python and data science, and you didn't able to learn the above, you will get a job of data analyst. And like that, you go to machine learning, you will get a job of data scientist, machine learning scientist, and so on. You go up to deep learning, you will get that job. And if you cross up to AI, you are eligible for all kind of job role in the market. Okay, this is for the student community. Okay, so you can use this data science in the step-by process in the organization as well, and you can get a different kind of job designation. You can get a different kind of research also, because at every stages we can do a different kind of research. Hence, follow this roadmap. Extremely important for all those who are very new to this technology, very new. And after this, what is the relationship between data science, machine learning, and AI? There are the 4 different words I'm talking constantly in my talk: data science, machine learning, deep learning, and AI. Many people think that this all 4 meaning is same, but they are wrong. The all 4 meaning is not same, it is different. Hence, it is written in this ladder, 4 different steps. And the roles and responsibility of this all 4 words are different. So in the relationship, if you want to learn AI, AI is the bigger umbrella. Machine learning is the subset of AI, and deep learning is the subset of machine learning. Okay. So in this diagram, if you observe artificial intelligence, as I told you, I'm having the habit to explain the technology from the name itself. Break the artificial intelligence into 2 parts: artificial and intelligence. Separate it. What is artificial intelligence mean now? Our duty in the company is that to provide intelligence to the systems artificially, to make an intelligent system. Simple and sober. Artificial intelligence means make such a system or machine which is an intelligent system and which behave like human, which mimic like a human brain. This is AI. That's why the name has been given artificial intelligence. And machine learning then is a subset of AI. Machine learning means world of algorithm. Although on the top end, guys, machine learning word is used in the market. But when you will actually learn machine learning concept, you will came to know inside algorithms are learning, not machine. Name has been given machine learning, but inside algorithms are learning from the data, and they are ready for the prediction. When algorithms learn from the data, they will get some experience from the data, how this data is there, what is the pattern inside the data, and they are ready for the future purpose for the prediction. Deep learning is the world of neural network. Okay, and these are the relationship. So artificial intelligence means to make intelligent machine behave like a human. Deep learning and machine learning from the data, and they are ready for the prediction for the future data. So these are all the meaning of this. So finally, if anyone asks you, you want to tell this answer. What is data science? Data science is used for cleaning of the data and make an insight from that. Whatever the clean data you are available after the output of data science, that clean data is given to machine learning and deep learning team. They are applying algorithm to the clean data. Algorithm will learn from the clean data and give the output as a model, which is used for the prediction. And then you can go for artificial intelligence. That predictions are converted into real-time actions, intelligent actions. That is the duty of AI. So these are the all 4 different meaning. It is not same. It is under one domain called as data, but the meaning of all 4 word is different. After knowing this is the most important diagram, and after that a few minutes explanation, I will directly go for the practical. Please concentrate on this diagram. This is the diagram which explaining how we people inside the industry executing any project. And I will explain in such a easy way so that any layman person can understand. Any layman. So please concentrate. In this diagram, there are the 3 blocks. Block number A, block number B, and block number C. A block is called as big data engineering. B is called as the world of data science, machine learning, deep learning, and AI, which is the hero of today's world. And the C is called as deployment. What is our today's topic? Breast cancer prediction using machine learning algorithm. So I am taking that example now. Always remember one thing. In the field of data science and machine learning, whenever we are doing any project, so first of all, we have to collect the data related to that project. So our today's topic is breast cancer prediction using machine learning algorithm. So for this topic, we need to collect the data related to breast cancers, patient data, past data we have to collect. Now I don't know that participant is having this feasibility to unmute themselves or not. I want to ask you people, from where we will get the breast cancer patient data? Can anyone tell me? Can you people unmute, or can either you can put a message in the WhatsApp, sorry, in your chat window, that from where we will get a breast cancer patient data? This is the need, because we have to solve that project. Our project name is breast cancer prediction. So from where? Someone's are responding from hospitals. Very good. Okay. Pathologists are there. So obviously, now I want to ask one question to you. If I will ask, go to your city and ask that cancer hospital and pathology to give the cancer patient data. They will give you data? Ask go hospital and ask them, give me cancer patient data. They will give you data? Yes, please tell me. Answer is no. No one will give you data. So before that, you will have a tie-up between your data company, data science company, and the hospital. We are having a legal tie-up, and after that, hospital is ready to give you data. Now, what you are going to do, guys? What you are going to do? Your hospitals are ready to give you data now. Now the responsibility of collecting the breast cancer patient data from the hospital, from the pathology, that is various sources, with the help of various tools and technology, is the responsibility of block number A in this diagram. And that block A is called in the company big data engineering. Big data engineering team is having the responsibility of collecting the data related to project from the various sources with the help of various tools and technology. And that's what it is called as big data engineering. So it's having the 4 major responsibility: collecting the data, storing the data, 2. Then third, maintenance of the data and security of the data, having the big data engineering. So they are collecting data and keep in the database of the company. And this data is called as a raw data. So remember, from the first slide, I'm talking about companies are collecting data for the different purposes. That data is called as a raw data. So here our raw data is breast cancer patient data. And it is up to us. During the project, we are deciding in what format of data we want breast cancer patient data. Either we want in terms of images, or either we want in terms of some values of that cancerous cell values. So it is up to us for that project demand. So today I am going to explain the project not on the basis of images. I am going to explain on the basis of some numerical values of the cell. So data is collected. Now what we are doing now, from here our responsibility start in the company. That is the part number B. In the part number B, very first thing is data science. So please follow my that ladder. First is Python language, then data science. So here first we assume that Python is learned. Now data science is started. What this data science team will do? They will read the breast cancer patient data from the database with the help of Python language. And they are going to do this 3 words: analyze the data, visualize the data, and interpret the data. These 3 different words is called as a data cleaning in the company. Data cleaning in the company. Why? Because it is extremely important to clean the data. Cleaning the data means what, guys? Suppose there is any error in the data and many missing values are present in the data. We need to treat that. That is the cleaning of data. Once you clean the breast cancer patient data, then you are giving that clean data to machine learning and deep learning team. That team is applying the algorithm to the clean data. So here one equation will form: clean data plus algorithm. And this called as machine learning. Algorithms are learning from the clean data, and the output of the algorithm is a model. And see, output of block number B is a model. Model means some mathematical equation. They are knowing that what hidden information that data is having. So companies are ready to pay a good amount of money if we are giving them a good model. Good model means prediction is good. Models are used for prediction, future data prediction. So we are having that model available of breast cancer. That model is having the information that if this is the characteristics of a patient, patient might not have a cancer. If this is the characteristics of a patient, patient might have a cancer. That information model is having. Hence, companies are ready to pay the money. So if we have tested our model is performing very good, then we are going for deployment so that common public can use that. So deployment team is cloud computing team, Amazon Web Service, Google Cloud Platform, or Microsoft Azure. So what we are doing, we are applying cloud computing to the model, or in other words, we are giving our model to deployment team to convert the model into real-time application called mobile app or website, anything, so that we user can use. So let's take an example. We have converted in the form of mobile app, and that app is available in the Play Store now. So common public will download the app, breast cancer prediction app, and that app will ask some input. You are giving that input, so that app output will be you are not having cancer. So in this way, we are using any project using the data science technology from end to end, starting from the project topic to the deployment process. So what in the project of machine learning, deep learning, and AI, one team is involved for collecting, storing, maintaining, and security of the data, that is big data engineering. After that, other teams are responsible for reading the data, analyzing, interpreting, and visualizing, is called as data analyst in the company. And visualization is always helpful, because with the help of visualization report only, we are giving explanation, presentation to stakeholder. So visualization is very powerful tool. So you should know data visualization. And after this, data visualization can be done in n number of ways, with the help of language or with the help of software tools, that is Tableau or Power BI. After that, we are giving the clean data to machine learning. And in the machine learning, this dialogue is very famous. Quality input gives quality output. So machine learning are getting the input from output of data science. Hence, there is an importance here. Data science team need to give a quality input to machine learning team. That is clean data. So machine learning algorithm will learn qualitatively, and output is quality output. So quality input gives quality output. This is the dialogue in the company. Other dialogue is also very famous. Garbage input gives garbage output. If you will not clean the data well, you will get a garbage output. And it is a very serious matter. If I am not going to clean the cancer patient data neat and clean with a seriousness, and giving the unclean data to algorithm for learning, algorithm will learn and give the output, but that output is of no use. It is hazardous. Okay, so garbage input gives garbage output. So finally, output of machine learning is a model which is given for the deployment team for converting into real-time application. And in our data science people life every day, we are giving a huge amount of time of a project to clean the data. You can see in this slide. Many people outside the data science field, they are thinking we people are having the ready-made algorithm available and applying, and game is over. No, we are putting our lots of effort to understand the nature of data, which algorithm is suitable for that, how to clean the data, and our majority of time is going for data cleaning, because on the basis of data cleaning, your algorithm will learn properly. Hence, in every project, 70 to 80% of time of a project is given for a data cleaning. So this is a data scientist life. Okay, and then after that, this is the very important, then we will go for our project. Any project in the world of data science and machine learning and AI, you have to follow this ladder. Compulsory. Business understanding. So business understanding means what is our project about? Our project is breast cancer prediction using machine learning algorithm. We are giving the huge time here. And I'm honestly saying in front of you guys, okay, I am expertise in the field of data science, machine learning, AI, and we are applying this technology in various domain. So today's topic is on genomics. This is the domain for me. It does not mean that I am having the knowledge of that domain. Okay. Hence, in the company, companies are hiring the domain experts. See, I am an engineer, guys. I am not from medical background. I don't know how the cancer is happening. I don't know how the brain tumor is happening. I'm not from the mechanical background. So I don't know how the automatic car is working. And I got that project. So what should I do? So I am having the knowledge of data science and machine learning. Then companies are giving the domain expert. So we people are sitting with the domain expert, And we people are doing a lots of quality discussion, how to solve this project and how to go ahead. So first is business understanding. Here business understanding, all people are sitting together, domain experts and a technological expert. After that, they are deciding to do this project, what kind of data is required. What kind of data is required to do this project. So suppose we people are doing the project of breast cancer prediction. So we have to decide to predict the breast cancer. What kind of data is required to collect from the patient? What kind of data? Either it is a numerical data, non-numerical, any kind of data, that is a discussion. Once data requirement stage is over, then data collection process is start. As I told you right now, data collection means from big data engineering. So they are collecting the data. During the data collection, so many error occur, manual error occurring. Okay, some human are doing the data entry job. They are not educated. They are just writing anything, and missing values are available at that time. So during data collection, error occur. That's why there is a need of data cleaning. So after doing the data collection of project, then we are going to clean the data. That is called as the data preparation. Okay, this is a life cycle of project. Once you clean the data, then that clean data we are giving for machine learning. That is called as a data modeling. Data modeling means giving the algorithm to the data. Algorithm will learn from the data. Output of algorithm is a model. So model evaluation meaning in the dictionary is test. So model testing, test the model working properly or not, giving the right prediction or not. If it is giving the right prediction, then go for the deployment for the common public, so that common public can use that. And after that, during the usage of common publics are facing any problem or not. Okay, so monitor that. And if they are facing the problem, so iterate the overall project cycle again to give the quality outcome. So this is the data science life cycle. Everywhere in the world, we are following the same steps, and in the step there is no change. The only thing is that you need to have that skill set requirement to do these projects. And after doing this, I have already explained these slides. Okay, so these are the tools and technology to learn the data science. So to learn the data science, one programming language, that is Python. Very useful, easy to learn. After learning Python, learn the mathematics. Mathematics, not complete mathematics, only 4 chapters, and I will let you know what are those. Okay, then learn machine learning algorithms and learn data cleaning. And domain expertise is optional. Okay, domain expertise, no one is perfect in the market. No data scientist knows all the domain. They are knowing only those domain, those they are involved in the project, and that too they are taking the help from domain expertise. So these 4 things you should know: Python language, machine learning algorithm, data visualization, and maths. And by knowing these 4 things, okay, then we can go for doing the research, either applying for a job, either you can open your own startup. So as I told you in my ladder, first you are going to learn Python, then you are going to learn data science. So this is the data science libraries you are going to learn. Only for the information I am showing this. Then machine learning, after learning data science, machine learning. So machine learning, this equation I told you, clean data plus algorithm is equal to model, and that model used for predictions. This is a machine learning, guys. So machine learning are training the machine. Machine means actually a system where algorithms are there and they are learning from the data. These are the 4 different types of machine learning: supervised, unsupervised, semi-supervised, and reinforcement. Due to the time limitation, I cannot able to explain the technicality of that, but just do remember, as a layman person point of view, that machine learnings are used for applying algorithm which is already made. You don't have to design also. Okay, and apply that algorithm and algorithm learning from the data. So these are the 4 different types of machine learning algorithms: supervised learning, unsupervised, semi-supervised, reinforcement. Today's our project we are solving with the help of supervised learning. Supervised learning means what? Some supervisor is there to train the data in layman term, and in the technological term, in the data, input and output both are available. Outputs mean the labeled data. Supervised learning means labeled data. Output must be present in the data, and algorithm will learn from the input output pattern. If this is the input, this is the output. This is the input, this is the output. So algorithm learn from that. So here also we are having the images as the input of dog and cat, but to every images during the data preparation, you are writing this image is of dog, this image is of cat. So that name cat and dat as per a label data. And this label data you are giving to the algorithm. Image is the input, to that image output is cat. So algorithm learning that, oh, this image is of cat, this image is of dog, this is cat, like that, input output mapping. That is supervised learning, output is present. And hence, we are predicting the same for the future purpose. So this is supervised learning. In the supervised learning, there are two types, regression and classification. If your output is in continuous in nature, it is a regression analysis. Output is discrete in nature, classification. Okay, these are the various algorithms. Okay, I don't know that I'm going fast or not. I'm not sorry. I'm not taking the feedback. Please let me know I'm going fast or this speed is good. Is this going above the head? Please let me know. Is this speed is good or I'm going fast or what? Please tell me. It's good. Thank you. Thank you very much. Okay. So these are the various algorithms, and as you know that it is not possible for me to go directly deep into that. Large amount of time is required for me for that. But just showing, if in future you want to learn, so this algorithm you want to learn in regression. Then classification algorithm. If your output is discrete in nature, yes or no. So today we are doing our project with the help of classification only. This cell is having cancerous cell or not, malignant and benign cell or not, yes or no. So when your output is yes or no, discrete in nature, apply classification algorithms. Okay, these are the various classification algorithms. Then after that, unsupervised learning. Unsupervised learning means unlabeled data. Output is not present in the data. Learn only from the input pattern and form a similar group. So here we are giving only the images of cat and dog. We are not writing in front of image, this is cat, dog. Give image to the algorithm. What algorithm is doing? Algorithm is learning from every image's characteristics, pattern. Okay, this image is having these characteristics, this image is having these characteristics. Both are having same or what? If both are having same, put both the image in same group. So finally, they are forming the two cluster, two group. One group image is of dog characteristics and one is of cat. This is unsupervised learning. Only learning from the input pattern, no output. Okay, and output of unsupervised is always a group 1, group 2, group 3. Group 1 is having the similar input, group 2 is having the similar input, like that. Then finally, these are the various algorithm of unsupervised learning. Then reinforcement learning, fourth type, agent environment learning. Today you people are the agent, I am your environment. You are learning from me. After learning, you are taking some decision. If the decision is correct, you will be rewarded. If the decision is wrong, you will be punished. So this is useful in the gaming industry, reinforcement learning. So finally, any machine learning cycle is working according to this diagram. We are having the data of the project. That data is divided into training and testing. Okay, first clean the data. Okay, data of the project, clean the data, divide the data into training and testing. That training data is given to algorithm for learning. Algorithm will learn from that. Output of algorithm is a model. That model we will give some testing data for validation. Validate that it is working properly or not. If you are confident that model is working properly, now see what is happening in this slide. We are going to give the future cancer patient data to the model as an input. And that model will tell us that this input data is having the cancer or not. That patient is having cancer or not. So models are used for predictions. In this way, machine learning working methodology is there behind the scene. Okay, in this way it is working. And after this, our drawbacks of machine learning, this is overcome by deep learning. Deep learning is the world of neural network. Okay, world of neural network. And in these are the various architecture. Might be, I don't know you'll understand or not, but it is a various architecture of deep learning, convolutional neural network, recurrent neural network, and so on. So anything, any project related to images and video are a computer vision. This is the one algorithm, convolutional neural network is one architecture. And this is especially made for the project of images and video. Projects of images and video is called computer vision. Computer vision means what? We are giving ability to machine to see something. Vision means eyes. From our eyes, we are observing the world. Same thing, machine should observe the world. We are giving the vision to the machine. That is computer vision. And it is the project related to images and video only. Similarly, The project related to text data. Right now I'm speaking in front of you. That is called as a natural language processing, giving the ability to system, machine, to understand, read, write human languages. That's what it is called as natural language processing. Alexa is the biggest example of NLP product. We are talking with Alexa. Alexa is giving the answer to us, real-time answers. So that architecture is called as recurrent neural network. And the moment you will learn the deep learning, then the door of AI will open. And the AI is having these three types, very interesting. And after this, I am going for the practical. So artificial intelligence is of three types: weak intelligence, strong intelligence, and super intelligence. Meaning is very interesting. As I told you, the dharma, religion of AI is to produce intelligent machine. So that intelligent machine intelligence, we are comparing with human intelligence. Compare machine intelligence with human intelligence. So in the comparison, you will find that today's as current scenario, that machine intelligence are far ahead with human intelligence in many terms. Faster mathematical calculation, memory, and so on. On the other hand, human intelligence are far ahead than machine intelligence in many terms. Emotion detection. We can feel the emotion, machine cannot. Taste of food. We can feel the taste of food, machine cannot. So there are so many things. So on the final conclusion, it is came to know that human intelligence win the race. Still, there is no machine in the world who can beat the human intelligence. That kind of intelligence is called as artificial weak intelligence. World is going through artificial weak intelligence phase right now. But scientists are very confident that in future they are going to bring such a machine which can feel the taste of food, which can feel the emotion, and so on. When the day will come, that means human intelligence is equal to machine intelligence. That is called as the artificial strong intelligence. Again, scientists are trying their level best and confident that in future a day will come when machine intelligence overtook human intelligence. That is called as artificial super intelligence. And many Hollywood movie you have watched that. That is the three types of AI. We are in the weak intelligence right now. After knowing this, so this is the slide according to me. Data science, machine learning, deep learning, and AI is an art of finding missing information from the, valuable information from the data, giving that valuable information to algorithm for learning, that is machine learning and deep learning, and converting that predictions into the real-time action with the help of AI. This is an art, and you people are the artists. And to become an artist, we should have certain weapons, resources to do this. So for that purpose, you should know Python language, you should know machine learning algorithm, mathematics, and data visualization knowledge. So these are the 4 mathematics chapter which I am talking about. All algorithms are made from this. These are school level mathematics, not go in deep, no PhD level and nothing, school level mathematics. Okay. And after learning this, okay, so we are having the various kind of job role in the market like this: data scientist, machine learning, deep learning, and AI, and so on. Okay, so these are the various job role. Data engineer is responsible for collecting data, data analyst to clean the data, machine learning apply the algorithm, and data scientist is the doctor of data. Okay, so these are all the job role, guys. Okay, so now we will go for today's our project. Okay, that is called as breast cancer prediction using machine learning algorithm. Okay, I don't know that participants are knowing the Python language or not. Just consider that I am showing you one demo how with the help of machine learning algorithms, Python as a language, mathematics knowledge, and the data visualization, we are solving the project of breast cancer detection using machine learning algorithm. So right now, if you remember the flow of project: business understanding, data requirement, data collection. These 3 steps right now we are not having because our data is already given in the open source internet. I have downloaded from there because I didn't went to hospital for collecting data. I have downloaded from the internet. Okay, breast cancer data. It is given for the open source. Anyone can do the research on that. So once the data is available, but in that what opportunity we miss? We miss the opportunity that what kind of data is collected. We cannot have a control now on that because data is collected and downloaded. Okay, so This is the breast cancer prediction using machine learning algorithm. Now what we are doing after collecting the data, we are going to read the data. So in the breast cancer prediction, we are observing the cell in our human body. One cell is there. So cell data is collected and numerical value of that respect to cell data is collected of the past patients. And they are written output also. This cell is a cancerous cell. So they are written 1, output is 1. This cell is having cancer. And if any cell information numerical data, it is not cancer because it is a past data. 0 is written. So we are having a data set of a biological cell numerical value. Okay. And output is written 1010. Any cell numerical value which is a cancerous cell, it is having written 1 as output. Any cell which is having the not a cancerous, it is written 0 as output. So 1 means it is a malignant tumor cell, which is a threatening, that is it is a cancerous cell. And the 0 means it is a benign cell, benign tumor cell. So what we are going to do, we are having the data available, numerical data of a cell. So all numerical values are available. So it will look like this. I will show also this one. See here. That cell mean radius is how much, mean texture is how much, mean perimeter, mean area, mean smoothness, all this. And honestly speaking, guys, I don't know what is this mean radius, mean texture, mean perimeter. For this purpose, we have to sit with doctor, domain expert. Sir, please tell me what is the meaning of mean radius, how it is affecting the output, how much importance it is. Because as a data scientist, I am just observing as a numerical value. But the domain knowledge behind that numerical value is domain expert. Hence, to solve any project in the world of data science, data scientist technical knowledge plus domain expertise with domain knowledge, combination of both will solve the very productive project output. So the thing is, guys, that we have collected data and we are going to solve with the help of machine learning and deep learning. So these are the various libraries available in the world of data science in the Python language. All these libraries are helping us to solve the project. Some libraries are used for mathematical calculation, that is called as NumPy, full form is numerical Python. Some libraries are used for data manipulation. Some libraries for data visualization. Whatever data is there, draw the graph of that and have an insight from that. So we are importing, that means running this cell. Okay, we are executing this. After importing the necessary library, we are loading the data. So as I told you, data is directly ready-made available. So we are loading the data directly from the library also, and I can import from the internet also. So the load breast cancer patient data, it is written here. This breast cancer patient data we are loading and giving to this variable called cancer underscore dataset. Now this variable is having the breast cancer data. After that, we are converting now this variable which is having the data to the data frame so that we can visualize the data like this. I'm coming on that, this one. See here. This line is responsible of converting that load data of breast cancer into data frame so that we can visualize the data in this format, rows and column. Data frame is rows and column format. I also, please guys, help me in understanding. You people are knowing Python or not? Because directly I'm explaining like a one-way communication. I should take a 1 minute break here to ask and interact with the participant. Are you people are completely new to this Python world, data science world, or having some gist about this? And I don't know if you are very new to this field and I'm explaining this, are you understanding or not? Be interactive with me so that it will be very helpful for all of us to understand what we are talking about. If you are very new to this field, so I will keep my presentation mode in a very understandable format. Okay, so one candidate said that very new to the world. Okay, just on, I'm not explaining any technicality here, how the algorithm is working, but on my normal word, at least you can understand this. You have seen the Excel sheet or not? Please tell me. Excel sheet. In the Excel sheet, rows and columns are filled, data is filled. So So this is the same. This is the data of our cancer cell, breast cancer cell. At least you can don't know the meaning behind the scene. At least this is in the front of you. Data look like this while reading the Python. At least please write down yes or no. Are you getting this or not? See, this is the data now. In this data, some rows are there and columns are there. That column is having the information of that cancerous cell, cancer. Okay. To understand the cancer cell, we need the help. Obviously, we don't know how the cancer is happening. So this is the data having the numerical values of the cell, that cell information. And at the output, we are having one target column. This target is called as, if this is the one patient input, guys. This is one patient input. This 17.33, this is some patient data. One row is some patient data. One row is one patient. And that is the past patient data. And here it is written 0. So in that past patient data, it is not a cancerous cell. It is a benign cell, benign, not malignant. 0 is written. We have collected that data, given the numerical value of that past patient, and 0 is written because that patient was not having cancer. We have collected some other patient data which is having the cancer, because they all came for the checking they are having cancer or not in the hospitals. We have collected that. So what is our duty now? We are giving this data to data cleaning process. After cleaning the data, we are giving to algorithm. Algorithm is going to learn from this data. How? Algorithm will learn if this is all the input. This all numerical values are the input. So this is the output attached with that. So input output mapping. Algorithm will learn from that. And you don't need to do design algorithm. It is already designed. You just ready-madely apply on that. So algorithm is learning from the data. After learning from this numerical data, if this is the input, this is the output. This is the input, this is the output. So it learning some pattern from that. When the input is 0, sorry, output is 0, and when the output is 1, learning from that. After it learn from that, algorithms gives one output that is called as a model. Now that model is very important for us, because that model is having that in what we can say hidden information. It has been formed from this data, that when the patient might be having cancer and when not having, that information model is having. And we are using that model for the prediction. So this is the data, okay, and we are observing that. So to read the first 5 rows of the data, we are running this instruction, okay, and this is the first 5 rows you are observing. Then similarly, the last 5 rows of the data I can write down here. Tail means last 5 row I am observing, okay. And then if I want to know all the column name of the data, this is the data set I have shown you in front of you. I want to know all the column name. Just I am writing that variable dot column. It will give all the column name. That's what Python is very easy, guys. Python is very easy. It is giving all the column name. If I want to know what are the data type of column, so just write down D type. It is going to give data type of the column. Data type means it is a numerical value, but it is a integer or float. So it is showing that all the data is a floating. Check in the data also. So this all the data is having floating, and I expect that you know floating meaning. Okay, this all we studied in the schools. So floating data types, all the data. Okay, and then finally we are checking the missing value is available in the data. As I told you, during the data collection, missing value might be present. So to check in the data missing value is there or not, we are writing these instructions. Check in any column missing values are present or not. After running this, every column output is coming 0, 0, 0. 0 means there is no missing values in that column. And now here I want help from you people also. Might be some here expert is sitting from the genomics background. Those who have observed the cancer cell before. These are all the data collected for that cell. What is the mean radius, mean texture? These are all column name. And on the value, on the basis of this information, doctor are deciding, previously they are decided that the cell is malignant or benign. This is the case. And now we have to solve this problem with the help of machine learning. We are just solving that. Okay, these are the cell characters. Absolutely, Pooja. These are all cell characteristics. And I am not from this domain. I am from the data science. So we, and suppose you are having that domain knowledge and I am sitting together for this project, and then we are deciding out of this column, which are the important column present here, which is deciding that cell is having cancer or not. So that process is called as column selection. Select the important column. Okay, these are all column name. Now we came to know in every column all information is present. No information is missing because output is 0. There is no missing information in all column. Then the next step we are going that if there is no missing information, then we are going for further data cleaning. And what is that further data cleaning? I will let you know. Okay. So once we came to know there is no missing information, so the next level of is that mathematical analysis of all column. That is called as this method, statistical analysis. Statistical analysis. Mathematical analysis means what? We have all this column, and in that column numerical values are there. So what is the average of that numerical value? What is the average of this column? It is written here, mean. Mean means average of mean radius column. What is the minimum value of that column? It is written 6.98. What is the maximum value of that column? It is written 28.10. So such kind of information we are gathering of that data set. What is the minimum value of column, maximum value, average, and so on. This information is helping us for analysis. And obviously domain expert is also telling us that what might be the information. But as today I'm saying in front of you, I'm not having the domain knowledge of this, but also I have run this project. Why? Because of the technological knowledge. I have just run this. I have treated the missing value. I have checked that nothing is there missing value. I have gone mathematical analysis, and so I have run the project without the domain knowledge. But you can imagine when domain knowledge will sit with me, we can improve the result. So I hope you are understanding now. In the world of data science means technology. When we are applying this technology in any domain, healthcare domain and banking domain, you can apply. So banking expert will be needed. So data science technology, when you apply in any domain for profit or business, so this number of teams involved to solve the project. So please tell me, are you understanding clearly or not? So that I can then go ahead how to show that how algorithm is applied. Understood? On the don't go on technicality, on the layman term, on the top data science, then domain knowledge is required for project. Are you getting, all of you? Or is this thing going above the head? Okay, no worry. So the thing is, guys, then once you analyze the data, then we are going for data visualization. Any column data you give to that library, okay, that library will show you the graph. From that graph, you can analyze that might be what the column is trying to tell us on the basis of graph. Okay, on the basis of graph. So these are the data visualization we have done on the cancer data, where we are having such kind of analysis. Okay, this is the graph, but it is a very big graph. Okay, so from this, if you can go for the smaller one like this. Okay, count plot means out of the total number of patient data which is having, how many patient is having cancer and how many not. So it is showing that graph. This graph is telling me that 200 patient, see here on X and Y axis, at least you are knowing this. I expect that you are knowing this, how to analyze the graph. See on the Y axis, count is written 0, 50, 100 like that. On the X axis, we are having the bar plot. So see the blue color on the X axis, 0 is written 0. So this patient is not having the cancer. And there are 200 around patient which is not having cancer. Past data analysis. And here one is having the cancer. So around 350 people is having the cancer. Okay, that is the past data analysis. So this graph is telling us, so at least during the presentation, we can show that out of the 100, let's take an example, out of the 100 patient, this amount of percentage is having the cancer data and this is not. On the basis of graph, you can tell that. And similarly, you can go for the further analysis of the data visualization, that this graph is telling us the relationship between all the input column to the output column. Mathematics is there. Relationship between all the input column with the output column. If the numerical value is more than 0.70, 70%, then both column is having the strong relationship with each other. That is the meaning of this. We are having the column in the data. We are finding the relationship of all column with respect to each other. Okay, we are comparing both columns and we are finding the relationship with some numerical value. That is called as a correlation. Mathematical terms, that is correlation. So we are getting this. Why we are doing this? Because we are identifying which column is having the highest correlation with the output column. But that highest number is giving me information that this column is having the highest impact on the output. This column is very important. If any column, input column, is having the correlation more than 0.7 with respect to output, because you have compared input column with output also here. So that column is very important. So that information we are getting without domain knowledge. Without domain knowledge. Hence we did that. And after that, this all are the part of data pre-processing. We are doing that, and finally, we are splitting our data into training and testing. Okay, after cleaning the data, split the data into training and testing. So we are splitting the data into training and testing here. This one. All the input is given to one variable, output is given to one variable. So input and output. I already told you input output analysis algorithm will learn. Then we will be dividing the data, as I said, into training and testing with the help of this instruction. Okay. Data is divided into training and testing. Then after dividing the data into training and testing, here we are applying algorithm. This is the algorithm, guys. And we didn't design this. This is designed by a year ago. We are just ready-madely using that algorithm without any coding also, directly calling the algorithm, this algorithm, SVM, support vector machine. Directly called, you don't have to write anything. Algorithm has been called and applied there. That's it. It's so much easy. It is something like using the app in your mobile. And you are very good in using the app with the favorite app, photo editing app. You are knowing that how to call the image from our mobile phone, that is import, how to apply the filter and all that we know. Okay. So this thing also is very easy. Just first time it will be hard. When I learned, first time I found it hard. But the day I will practice, it gives so much easy now. Just ready-madely call the algorithm and ready-madely give the data set to the algorithm. So we are giving the training data set to this algorithm here. See here, this line, we are calling the algorithm, and to that algorithm we are giving our training data. And this line, which is I'm selecting the line number 3, it is called as algorithm is learning from the data. Because we are written algorithm.fit xtrain ytrain. Xtrain is the input, ytrain is the output, training data. So we are giving training data to algorithm. Algorithm is learning from that. After learning, algorithm will give as output model. So this is the model, line number 4, and this model is used for prediction. So testing data we are giving for the model for prediction, and now output is coming in terms of accuracy. So 0.93 is the accuracy of our algorithm right now. Without any domain knowledge, I just run this project. 0.93 is the accuracy coming. But I need to consult with domain expert also in order to validate this result. Okay. 0.93. So this is one algorithm I have run. There are many classification algorithms. I have run all classification algorithms. This is one algorithm. This is another algorithm, logistic regression. I have applied logistic regression algorithm on our same training data, and I have checked the performance. So it is coming 0.95. Previously it was something 0.93 and it's a 0.95. Improved. This algorithm is working better on that. Okay. Similarly, I have applied various algorithms here, KNN. Now I have applied KNN algorithm. Now 0.93, which is coming less as compared to previous algorithm. Then we are going for neo bias. Okay. 94% accuracy. And within this all instruction, you don't need to mug up. You don't need to do anything. Directly copy from the internet also. but you should know how to apply data on this algorithm. That's it. That's a talent, skill, and company are hiring for you for this purpose also. You are, because in the company also, we as an employee, I didn't design this algorithm, designed by scientists. I am using that algorithm. Every company is using this. So the thing is that you should know the ability how to apply the data on the following algorithm, and you should know how that algorithm is working. That's it. Okay. And then you can go ahead for the further analysis. So all algorithm we have applied, and this algorithm is giving 94% accuracy, decision tree. Okay. And then random forest 97 and so on. Might be I'm not why I'm going very fast, because you people are not aware of this all algorithm. I'm just showing you the result, accuracy result. Okay, so we are getting the accuracy result of all algorithm, and finally you can conclude that after applying 8 algorithm, this algorithm is giving the highest accuracy. So you can conclude, and you can present a paper also on this file which I am having right now. You can present one international journal paper that on the breast cancer prediction when we apply all the classification algorithms. So this is the result. When we will give the same data set to another data scientist, so that data scientist might be having different approach, which I have run right now. So you can imagine one data set, just give to the 10 data scientists in 10 different rooms. All 10 different results will be different, because everyone is having their own approach. It is same thing. If my stomach is paining, I will visit to one doctor. That doctor will diagnose me and give me medicine. I will again come out from that doctor lab. I will visit to another doctor. That doctor will diagnose me with a different approach. So it is the same thing. Okay. So in this way, guys, we are doing the project in the company, right from starting the business understanding, data requirement, data collection, then data preparation, and so on. Missing value treatment, data visualization, and everything. And then finally, we are applying the algorithm. And by applying all algorithm, we are giving the final analysis. Okay. We are also validating the model, guys. Okay. That is the higher level approach. Right now, I'm not going on that, but that is a process. Output of model, we are validating, checking it is working properly or not. Should we go ahead with these results or not? We have to validate that. So the thing is there that I have run this file with the help of Python language, and we have applied the various machine learning algorithm to this. And finally, what we can say that, as I told you, that apply the data science and machine learning technology into the various domain, and you can get a various kind of job designation like this: data engineer if you are interested for the collection of data, data analyst if you are interested of data cleaning and finding the insight, then data scientist, doctor of data, and so on. So these are all the case, different job roles. I have explained to you what skill sets are required for Python language, that for mathematics knowledge, algorithm knowledge, and data visualization. That's it. You can apply for a job in company, you can do research, and if you are having the very good knowledge, you can open your own startup. So this is the all case. Future means different application of the data science, different applications. Okay. So you can apply in the medical industry in the n number of projects, not only cancer detection, brain tumor detection. And let me give you one information. Healthcare is the now #1 domain in our data science, because there are many research projects is going on, and if you succeeded, believe me that many life is going to save in future, because we are going to detect and predict the deadliest diseases in advance only. Why cancer is very deadliest? Because it is getting detected at the 4th stage and 3rd stage. And if it had been detected at a very earlier stage, the life of that person can be increased. So that can be done with the help of AI. And this AI is having that number of application, speech recognition, robotics industry, and other applications. Okay. So this is the all Google driverless car, 1,000 application, guys, 1,000. Okay. So you are surrounded by data science and machine learning application every day. And finally, what is our challenges in this field? Before concluding, the challenges is that as up to this session, you came to know very clearly, to do any project in the world of data science and machine learning, we require data. And if we didn't get quality data, then it is a headache for us. if you didn't get quality data. So getting the quality data is first challenge. Second challenge, we are not getting the skilled data science professional which knows how algorithm is working, how mathematical aspects are there and everything. Many a time startups are not giving financial support to their research and development team. Many a time we are not getting domain expert. Project is available, project name is available, but domain expert is not available. So project is not going ahead. So there are many challenges. And by overcoming these challenges, what is the takeaway of today's session? This is the takeaway, this diagram. For solving any project from any domain, flow is same. Domain might be anything, flow is same. You want to suggest some project name that is called as business objective. To complete that project, what kind of data is required, note down, collect the data. Okay. During data collection, many error can occur. After collecting the data, now it is up to us. Everything is discussion board. In what format you need to collect data? You want image data or you want Excel sheet data? What data you want? What kind of column you want? Numerical, non-numerical, what? Everything is the part of research in the starting phase of the project. Once data is collected, then you are going to clean the data with the help of Python and what the four thing I have told you: Python, algorithm, data visualization, and maths. Output is a model. Output you are giving to algorithm. Algorithm learning from the data. Output is a model. Model is used for prediction, and then it is going for deployment and monitoring. So this thing we have learned. Okay. So data science is the field where it is used finding the hidden information, valuable information from the data, and that valuable and hidden information is used for the business decisions, for the growth purpose. And you people are hired for the same. Okay. And various job roles, I have explained what are the skill set required, what are the applications, challenges, and so on. So thank you very much, guys. That's all from my end. If you are having any doubt, now I am open for the doubt solving session. I can guide you whatever I can. Thank you very much. And thank you, Pankaj sir, for organizing such a wonderful event. And now I am open for the doubt solving session. You can reach me in this contact information if you want the knowledge about Python, machine learning, deep learning, and AI in very easy way. You can connect with my LinkedIn. You can connect with my email ID. Thank you very much. Now I am open for the doubt solving session.
Thank you, Mr. Arpit, for such an intriguing and elaborate talk. So the session is now open for Q&A. You can type in the chat box or you can raise your hand so we can shift you to the panelist.
Some student is asking doubt, what is the public repository? You can go to Kaggle website also, Kaggle. There you can just write down in the Google, breast cancer data, Kaggle. Or directly if you write down this word also in Google, breast cancer data, same data is available in various repositories. You can download from any one of them. Kaggle is very famous. Kaggle is very famous. Or even though on the today's project, I have taken the data from the library itself. As I said, Python is a very powerful language. Now in that library, data sets are also ready-made available.
Yeah, good morning, Mr.
If you want, you can email me. I will give the data also.
Good morning, Mr. Arpit.
Yeah, good morning.
Since you have worked on AI and ML and you have a rich experience on this one, can you suggest something like if somebody has to learn machine learning or machine learning is a course which need to be taught to the graduates? What should be the content of that course? Because the courses which are run by different universities, there are so much variations. And sometimes it's difficult to understand what should be the content for the first machine learning course to the students.
Yes. See, as I told you, I am working as a mentor across the world with various universities also, where I am responsible for designing the curriculum as well. So see, this is the machine learning curriculum already designed Okay, what should be the first step, what should be the second step, and in the step-wise direction I have designed, and it is appreciated and it is selected for IIT Guwahati curriculum as well for one program. So this is the data science, machine learning, deep learning curriculum. But as I told you, whenever if you want to teach machine learning directly, don't go directly. My strong suggestion, follow this roadmap, because I am involved. I know what are the challenges other universities also facing, their student facing, because they are not following this roadmap. So you want to go designing the structure in this way.
So
First,
can you share your curriculum that you have designed with us?
Yeah, I can share that. I can share that. And the curriculum, all the curriculum designing is by following this roadmap. Okay. First, Python. After Python, we are introducing data science. After introducing data science, then machine learning, because machine learning required the output of data science. So hence, we cannot directly launch machine learning, step by step. So this all, whatever I'm showing in this slide, all curriculum are ready.
Right. So I'll drop you a mail, and in the reply, if you feel suitable, you can send me your designed curriculum.
Sure, myself. Because why? As you can observe from this slide, step-wise job designations are also available. You can publicize like that also. Up to this course, this might be the outcome. Up to this certification course, this might be the outcome. So this is the road-wise and quality roadmap. As sure I will share. I mean, so you just mail me. I have shared my contact details also.
Right. Yeah.
I'm available on the WhatsApp also on the same number. Anyone can approach for any kind of help. I will be happy to help you out.
Thank you. Thank you.
I don't know. I hope the session is enjoyable. I don't know, might be it's going above the head or what, high-end technology, but I tried my level best to explain in the layman term. I haven't used any technical term.
No, it was good and quite informative. And I can understand the limitation because the field is very vast and you have to deliver within 1.5 an hour. So it is a challenging job to deliver such a vast thing in a minimum amount of time.
Yeah, sir. Thanks for that. And for any update in the field of AI ML, you can connect with me in my LinkedIn, and also you can connect with our company website called AI Beehive, so that you can get a upcoming informations as well. Okay, this one AI Beehive, you can connect there as well.
Good afternoon.
Yeah, please.
Yeah, this is Vamsi. Today's session was enjoyed a lot. My thesis is also working on machine learning itself.
Okay.
I'm searching how the Python works on my stock market. My project is on stock market prediction using machine learning. I don't know about what to do, but today's session it was very helpful for me. Thank you very much, Arpit.
And I would like to give one information. Already we have solved the project, but it is not an industry project, it's an academic project. Stock price prediction using machine learning. We are having that.
Yeah, Arpit, can you please share that project?
Yeah, I can share you that project.
I will drop a mail for you.
Sure, you can drop me mail. I am having already that project executed. Stock price prediction using machine learning approach. Already having that.
Yeah. Thank you very much, Arpit. Very nice information. Thank you a lot.
Yeah, thank you.
Good afternoon, sir.
Good afternoon.
Sir, I am Kedar Deshpande from Nasik. I was just thinking what to do about do data science. I'm a mechanical engineer. But then when very initially when I watched your program, this session, my all doubts are clear. And now I will be planning to move in data science work field.
Obviously, Kedar, you do one thing for your kind information. As I know, data science is everywhere. You just watch this video after my talk. You will be fascinating by watching this video. As you told, you are from mechanical industry. Go and watch this video, recruitment car. It is a Volvo company product, AI product, where car is only recruiting its own employee. No human. Car is recruiting his own employee by taking the interview. It is a 2-minute video. Very motivating and fascinating for all mechanical engineers.
Okay, sir. Thank you, sir. Thanks a lot.
Recruitment car. Yeah. You can have a wonderful career in this. Can contact me anytime.
Yes. Thank you, sir.
Thank you. Anyone, those who want any generalized doubt also, not necessary technical doubt. Those who are planning to learn data science, machine learning, deep learning, how this is impacting different domains. I tried to explain in a layman term. The doubt, guys, you can unmute or. I think no doubt Pankaj sir is present. Pankaj sir, are you available?
So actually, Pankaj sir had a meeting with director sir, so he had
Okay.
to left the meeting.
Okay, okay. So shall we wind up the meeting or I have to wait if there is no further doubt?
Yes, let me check the chat box if there are any doubts.
Yes, yes, yes. Please, please.
So there is one question that which chapter we have to study in the stats.
Okay, statistics.
Yes.
Actually, the whole statistics we have to learn, but three thing. Descriptive statistics, inferential statistics, and hypothesis. These three main. Descriptive, inferential, and hypothesis. Very important. That one instruction which I run, the mean of that column, minimum value of that column, maximum value of that column is a descriptive statistics only.
So any other question or doubt regarding this session? Please type in the chat box or you can raise your hand.
Or you can connect with my LinkedIn also. No worry, guys. I will be happy to solve their doubt anytime. You can connect via any one of the contact board. This is my WhatsApp number, email ID, and LinkedIn ID.
I think there are no any other doubts. So I would like to thank you, sir, for sparing your time and giving such an informative talk.
Thanks.
Thank you, sir.
Thank you. Thank you very much.
So for all the participants,
One more question if
yes,
there. I am observing. Mahesh Kumar is asking, do we need to normalize the data? Yes, yes, it is a part of data cleaning. I haven't gone through that technicality because participant might not understand. Yeah, Mahesh Kumar, we have to normalize the data to bring in the same scale. Okay, thanks.
thank you, sir.
Thank you.
So for announcement for the participants, the next session will be at 3 o'clock by Dr. M. Muthuraman. So please join on time.
Hi, Pankaj.
Monica, you may start.
Okay, thank you, sir. Good afternoon, everyone. It is a great pleasure for me to introduce our speaker for today's talk, Dr. M. Muthuraman. M. Muthuraman was born in Chennai, India in 1980. He received a BE degree in Electronics and Communication Engineering from the University of Madras, India, and MS in Digital Communication in Christian Albert University, Kiel, Germany. PhD degree in Biomedical Engineering from the Technical Faculty and Department of Neurology of Christian Albert University, Kiel, Germany. In 2010, he joined the Department of Neurology, the University of Kiel, as a postdoc. Since December 2016, he has been with the Department of Neurology, Johannes Gutenberg University, Mainz, where he is an assistant professor and the head of the department, Biomedical Statistics and Multimodal Signal Processing Unit. His current research interests include mathematical methods for time series analysis and source analysis on oscillatory signals, biomedical statistics, connectivity analysis, multimodal signal processing and analysis of EEG, MEG, fMRI, and EMG, structural and network analysis on anatomical MRI and DTI, functional network analysis on PET imaging, machine learning and deep learning, network analysis on proteomics and genomics data, RNA, mRNA, and spatial transcriptomics. So please join me in welcoming Dr. M. Muthuraman to address the session on translational models in neurology. Thank you, sir, for coming. Over to you.
Thank you. Should I start?
Yes, Muthu, good afternoon and good morning. I think it's still good morning in Germany.
Sorry, I can't hear you very clearly. Should I start or?
Yes, you can start sharing your slides.
Okay. You see the slides, right?
Yeah, we can share and see the slides, yes.
Okay, so good afternoon, everybody. So my topic today would be translational models in neurology. And as already given a good introduction, I take care of the group here in Mainz for biomedical statistics and multimodal signal processing. And in this talk today, we will talk a lot about brain, human brain, and also mice brain. So that's why I would start with a introductory slide on human brain. Probably some of you are not familiar with this. So we have in the brain several lobes, we call it. These are separate parts in the brain which are responsible for certain tasks. So if, for example, you see my laser pointer here, and I want to point this laser pointer in the front of my brain, then I can just explain you what happens exactly in my brain for this particular task. You have the motor cortex, which is between this frontal and parietal lobe, which is actually doing or any kind of movement with the hand. And the parietal lobe is then giving you the orientation in which way I should move my hand. And the occipital lobe is then giving you the information of what I'm gathering from my eyes and then where I should point this laser pointer. And this temporal lobe here is then giving you the information which you want to store for using it later. So the memory part of your brain is mainly here. So for the next time, if I want to use this laser pointer, this then, which is stored in the memory, then directly being replicated. The red part is called the cerebellum, which is a very important region for all the motor tasks. And then you have this pons and the spinal cord connection, which is shown in green, which then gives the information to the spinal cord and to the muscle, and then we have the information in the hand which I need to move. Of course, this brain is then mainly has these lobes, and then each of these lobes can be also take into account several waves, or we call them oscillations in the brain. The low frequency oscillation is then the delta, which is from 0 to 3 hertz. That's the slowest. And this you can see when you're in asleep in most part of the brain. The theta frequency is from 4 to 7 hertz. And this you can see when you are certain task, for example, concentrating, then you can see it in the frontal lobe. The alpha frequency oscillation is then, which is from 8 to 13 hertz, which you can see mainly in this occipital lobe. And the beta is from 13 to 30 hertz, which is the frequency, and where you can see this mostly in the motor part of the brain. And the gamma is then, which you usually can record from the temporal lobe or the memory regions, which is then above 30 hertz. So now we've looked into the brain and also the oscillations which you can usually find in the brain when you record electrical or magnetic activity from this human brain. So now I would move to the next part of the talk, where I try to give you an overview what we do in our group and how we can link this translational models. So for building up this, we call this a platform so that we can always test this in humans and in mice with different modalities. Modalities means, for example, here the functional MRI, which can be used to measure the bold activity, which means just the blood flow in your brain, or the structural data, which is also giving you information, either how thick or how thin is your part, certain part of the brain, or you can also do something called tractography, which is then giving you the information from how one part of the region is connected to another part. And this you can do directly in human and also in mice brain. And additionally, we also record neuronal signals, meaning direct recordings from the brain, how what kind of activity is present at a certain state when you're doing a task or you're resting. So these are the modalities what we generally use in our group. And then what we try to do is we try to identify network changes, meaning in the brain, as we looked in the previous slide, there are several regions in the brain, and how do they communicate between each other? So this either can be a flexibility, meaning that from a resting state, for example, when you're sleeping, to going to a task, when you want to do a task, then these regions reorganize as shown here in this picture, and then it flexibly change their communication between the regions, and then that's why we call this flexibility, depending on the state. Or you can also directly between the regions with these arrows try to find how good they are communicating, or over time how this changes over time. Or we also use coupling measures. For example, if we record the brain and also record the muscle activity from the arm, how these two signals are coupled. So then you can use this coupling measures. These are then identifying network changes. And then what we want to do is a so-called model framework, meaning that once we have all this data and we have analyzed it, we want to develop a model. Either it can be a state space model, or structural equation models, meaning that we know the input. For example, if we have a patient and a healthy subject and we want to classify these two groups with the data and the analysis what we did, we can have this as an input and then try to predict the output, whether it is a patient or a healthy control subject. So with this possibility of having these models is that once you develop these models, then you can use these models again to have individualized prediction, meaning that we don't want to use these analysis for a group of subjects, but we want to also use these models what we have built, for example, in this deep learning architecture, and then try to predict one single patient when we have a new data set. How do these data can help us predict whether this is a patient or a healthy subject? And using these individual prediction, once we know that we have a patient data or a healthy control subject, then we can use this for therapeutic option, because we are working here with the neurology department. So we want to also have the possibility to give therapy for these patients. And we use different type of therapeutic options. We call this invasive or non-invasive intervention. Invasive means here as shown with a electrode. We, for example, in the Parkinson disease patients, we try to stimulate the brain so that they don't have this shivering in the hand. That's something which is invasive because we are doing an operation and putting an electrode in the brain, or a non-invasive stimulation here shown in picture, that you have a transcranial magnetic stimulation on the top of the head, and you specifically stimulate a certain part in the brain so that you can have kind of a therapeutic option for those patients which they don't need any invasive. We can also use this non-invasive, or we try to always check this with mouse models, for example, using a diet, as shown here in this picture, that using a diet we can simulate basically what's happening in the patient groups. I will give you some examples later. Or we can also prescribe certain lifestyle modification for patients, for example, daily activity, what they should do based on all these analysis. Or we can also give recommendations, for example, for mental health or weight control. And we also look at behavioral parameters, meaning how good does the mice move in a certain situation, how does the mice react in a certain situation when we give this specific diet for that mice, and then see how good they are in relation to the patients. So this is the platform what we use, and these are the different steps what we use in order to achieve this translational biomarkers, we call it. And first I then now go on to giving you certain examples for this. Always I either start with a human brain and then go to the mice example. So first I give you also more details about this deep brain stimulation. So here in this picture, as you can see, this is one hemisphere of the brain, and here is the deep brain stimulation electrode. Like I said, we open the brain and then have the electrode implanted. And what we do is that we have the possibility to record on the top of the scalp, meaning beneath the hair or on the skin. Those are EEG electrodes, or we have the capability also directly to record from the deeper regions in the brain, which is, of course, usually not possible when you don't do such an operation in the patients. And we can simultaneously measure muscle activity from the arm. So here again, some specific activity what we can record. when we can record the surface that's from the EEG, the EEG activity would look like this, which doesn't give you any particular frequency or oscillation like what we talked about. It looks like noise, but we have activity beneath this activity, meaning frequency activity beneath these surface recordings. Here is the axis in time, and here is an amplitude. Or we can also record local field potentials, meaning we can put a strip of electrodes here called the electrocardiography and then record directly from the top of the brain. And these are, of course, having a better amplitude compared to the surface because we are directly on the brain, or we can also directly record from the electrode itself. It can be local field potentials or single neuronal firing, meaning we can also record single neuronal activity from each of the neurons which is neighboring to this electrode, or we can record simultaneously the kinematic signal, meaning the muscle activity from the arm. And here are some more examples what can be measured from these signals. From these three signals, we can look, for example, coherence, meaning the EEG activity on the brain and the muscle. We then look at the coupling. Coherence is something like correlation in the time domain, but coherence in the frequency domain, and you have specific frequencies. Or we can look at amplitude coupling. For example, how does one frequency, let's say the beta frequency, which is I told earlier in the motor cortex, how does it couple with an high gamma frequency? That's, for example, the phase amplitude coupling, and you look for frequency to frequency relation. Or we can band pass filter this particular signal and then look at the burst activity. How do you see certain frequency bursting activity? Or you can directly look at the power spectral density of these signals and look where is the highest power. Or you can also look in time and in frequency, where do we see higher power, like these red regions or red time intervals and frequencies have a higher power. So these are just, again, methods what we can use from these recordings, what we record from the patients. And using this, we can also not only do electrophysiological recordings, we can also go for imaging biomarkers, meaning once this DBS electrode is here, so this is how the electrode would look like. Let's just give you an impression how big is this electrode. Is then about 7.5 millimeters, which is extremely small. And then we can also look, when we stimulate the brain, what amount of tissue are we stimulating. So this is how it would look like. And then we can look for several connectivity fingerprints, either, like I said before, using structural connectivity, how this particular part in the brain, when it's stimulated, how does it connect to several parts in the human brain. Or we can look at different stimulations and then look how do they isolate with the different fibers, like this diffusion tensor imaging, meaning one particular region, how do they connect to another region, we can isolate them. Or with the fMRI, functional MRI, we can also look at functional connectivity. For example, some regions have higher and these blue regions, for example, have lower connectivity. And then based on all these connectivity fingerprints, then we can use either a clinical scale, what we get from these patients, and then try to associate or even try to predict. So our final aim would be to have individualized therapy for these patients based on all these biomarkers. So now we have looked into the electrophysiological biomarkers and we have looked into the imaging biomarkers. So now I start with examples. As I said before, we look into Parkinson's disease patients. these are patients who have problems, like for example, starting with the hand, that they having a shivering or say movement involuntary, which means that they don't want to move the hand, but it automatically does this, so that, for example, they cannot do any daily life activity of drinking a coffee, for example, because they have this arm all the time in movement. So the therapy is then what we are doing is this deep brain stimulation. And simultaneously, what we do is, as shown here below, we record the electroencephalography. And these blue dots here on the top of the head are these EEG electrodes, which is placed on the top of the scalp. And then we measuring on the top of the head, but we want to look inside the brain where this activity is coming from. So what we do is something called source analysis, so that we identify the source in the brain from where this activity is coming. And we can look into the whole brain, or as I told before, that some brain regions are very important for this movement in the hand. So we looked into specific regions in the brain which are specifically important for the motor part. And for example, in the cerebellum and this subthalamic nucleus is where we have this electrode implanted in these patients. And we looked into something called cross-frequency coupling in each of these regions. So what we do in these patients is we stimulate at a certain frequency, which is 130 hertz, for example. And then we want to know how does this stimulation modulate other frequency oscillations like brain waves, what we talked before in the brain. So that's why we do this frequency to frequency, and then we are stimulating at 130 hertz and see what's happening in these regions at certain frequency ranges. And what we found is when we look at this amount of tissue which is activated in these patients, we can then identify certain markers, which is then identifying or giving us the information that when we stimulate these patients, we are modulating certain oscillations in these motor regions, so that this tremor or this shivering in the hand is not present in these patients. So what we can achieve by this is we understand how does this deep brain stimulation works, and we call these oscillatory proxies because we are looking at oscillatory signals in this human brain. So now we looked into the human brain. Now we can directly transfer this to a mouse brain. So in the mouse brain, on the left side, we also record directly from the motor cortex and the premotor cortex. So these are again regions which are very responsible for this motor part. We record in the mice. Of course, it's possible to have much more electrodes and much more detail. So here you can see the electrodes, these 4 strips, which is placed in this particular region and in another region in the premotor cortex, how these electrodes are placed. And then the nice thing about when we translate these from the human to the mice, we are able to record not only this kind of local field potentials or electrophysiological activity, we can also record these spikes, meaning how do they fire, the neurons firing in the mice in this particular region. And then what we do exactly the same way, we then compare the power in these 2 regions and see whether at a certain frequency, let's say beta, or here we look at 3 different gamma frequencies, whether there is a difference in power. So as you can see here, that in beta, for example, there is a reduction in this premotor cortex, the power, and this is also reflected in several layers in the brain. So in the brain, what we record is on the top of the scalp, and we don't have the information of each of these layers in the brain. from the human part because we are just measuring on the surface. But in the mouse or in the mice, it's nice because we get this detailed layer information because our brain is then layered with specific different tasks or activity, which we see here. So here are shown for 3 different layers. And again, we see this reduction in power. So this is what we see in the human part. We can directly translate. That's why we call this a translational biomarker, because we want to translate and see directly how it happening in the mice brain, so that we can understand even more detailed information in the layers. So that's why we call this translational biomarkers. And second information what I wanted to show is in another study where we also record in 4 different parts of the brain. So this is all in mice, and this is all regions which are involved in, let's say, when your brain it has stress, then all these regions are involved. And we were able to record all 3 regions simultaneously, and then we're able to look at the connectivity between these regions at several different frequency bands. Because the idea behind it is, again, I show here for each of these regions, these white parts just means those are the electrodes which are placed in each of these regions. So for example, this is the prefrontal cortex, which is in the front part of the brain. These 2 are regions which are in the temporal cortex, which is very important. All these 3 regions, which is very important for the memory formation. So when your brain it has stress, then we can see all these 4 regions play a role, how the stress is being kind of looking from the brain side, how do they react to this stress, and then what are the different regions and how do they connect with each other. So here is that you can see the thicker the arrow is, the higher the connectivity, and the color just represent from one single region, how do they connect to the other regions. As you can see that only the ventral hippocampus to BLA, there is no connection because we were only able to record 3 regions simultaneously, but all the other regions, as you can see, there is very large information flow or causality between these regions, which indicates that there is a lot of information happening in the brain, and these can be resolved in the mice because we are not able to look directly in the human brain by putting electrodes in 3 different regions. Here, there is a possibility that you can do that in the mice so that we get additional information from each of these single regions. How do they communicate between different regions in different frequency bands? Based on this translational biomarkers, now we move on to a different translational biomarker, which is usually used for earlier, whatever I showed is for Parkinson's disease patients, those models what we use. And this model is specific for multiple sclerosis patients because in those patients, specific parts in the brain are, we call it demyelinated, meaning that the information flow, how from one region to another region does not go in the proper way. They are scattered. So in that way, you can know these are MS patients, and we want to translate this in a mice model. And in this mice model, we give them a cuprizone diet, which is a specific diet which is given for the mice. So this is when they are at baseline. So then we don't give them any particular diet. But then when you give them for 6 weeks, this particular diet, then you can already see the change in the demyelination. This is an histological slices that these are becoming thicker. And then when you give them again normal diet, another 6 weeks, then you can see again they come back to the normal structure, which is seen before here. So this is just looking at the structure in the brain when you give them this specific diet in the mice. And you can do similarly in the mice also this diffusion weighted images where you can look at water molecules, how they travel in the brain. We divide the brain of the mouse brain into several different regions and then look at their connectivity between these regions and then develop certain network measures. Here I'm giving you an example, something called clustering coefficient, which simply means that how good the brain is connected. Cluster means that how good they are communicating with each other and how good they are clustered. And when you look at this connectivity over these time windows, so in the beginning, the clustering is very low. And then when they are having this diet, the clustering goes up. And then again, it comes back to the baseline after this remyelination, meaning when we give them this normal diet. So this is another way of representing that each of these uh, methods can identify these changes in the brain. So this happens mainly in the MS patients. So we went one step further and then looked at, for example, in the dynamic network changes, meaning over time, what's happening after weeks. Using this information, what we did is in human subjects, we looked into a specific region in the brain, which is called here, which is highlighted in the small red regions in the healthy controls and in the MS patient, and translating it to the mice brain. Here is when it's in control. Here are, like I said, the demyelination and then remyelination after giving the normal diet. We looked into this region called the choroid plexus, which is a very important region, which then gives you how two different hemispheres in the brain, how they are communicating with each other. And in the patients, like you can see here, the volume, how big is this choroid plexus, is increased in the MS patients, shown in red. In green is then the healthy controls, that the plexus volume is increased. And this is translated directly to the cuprizone mouse model as shown here. This is baseline when they have a normal diet. This is demyelination. There is an increase in this volume, and again going back to the normal remyelination, that where we give them the normal diet. And then we also replicated this in another mouse model where we can again see this increase in this volume. So all these two mouse models and in the human, we see this clear increase in the volume. And what we also want to find out is not only just the increase in the volume, but we want to also know whether this is related to the clinical scores in the patients. So this is what is shown here in baseline when the patients are diagnosed with multiple sclerosis. This is the volume. These dots represent each single patient. And here is a score, which is a clinical score for these patients. And you can see that they have a positive correlation, meaning when the patients are highly progressed in the disease, then they have a very high score of 6 or 7. And this is when they are in the beginning of the disease, that the volume is lower. But when they progress, you can see that the volume increases. So this is another indication, not only at baseline, but after, when we follow up the same patients after 2 years, we also see this increase in choroid plexus volume and the correlation to this clinical score. So this gives us again evidence that it's important to always translate the human findings, what we see also in mice, because we get more detailed information, and then try to always look whatever we cannot test in the human, we try to test it in the mouse models. And looking into this, now we will move on to First, we looked on the Parkinson disease models. Then we looked into multiple sclerosis. Now I'll show you a few slides on the aging biomarkers, because aging means that the diseases which are in this particular study, we call them mild cognitive impaired patients. So these are patients who have cognitive impairment, meaning that they cannot perform in a certain task. If you ask them to memorize a certain information and then they need to try to replicate it, it's not easy for these patients. And there also we look at frequency bands, for example, the delta frequency, and look at specific regions in the brain, and then look at their communication. As you can see, in the cognitively impaired patients, the dashed lines means the connectivity is lower. It means there is not good communication happening between these regions. In the healthy controls, when we compare, there is good communication. That's why it's shown with a bold arrow. That means there is good communication going on. That's why they are not cognitively impaired. Or we can look directly not only with the electrophysiology, we can also look at fMRI, or in this case, another method to look at association. How does this metabolism in the brain, how do they relate to the neurodegeneration? Because all these diseases are, the brain is degenerated. So you can see here, this is in the brain, those red regions are where the degeneration is higher, and in the blue regions are where the degeneration is lower. So you can directly relate to this clinical status, or we can also use this measures in order to identify the age of these subjects. So in this huge cohort where we looked at the volume, like I said, how big is this particular region, and here we looked at the cortical volume, and here are females and male subjects. And as you can see here with the clouds, there is a clear difference between the male and the female in the volume, in the cortical volume in the brain. So the male have a higher cortical volume compared to the females in most of these regions. And here is a table which shows we have done this in 816 subjects, and the age range is from 40 to 89. And these are different diseases which we used in this cohort. For example, the mild cognitive impairment, Parkinson's disease, dementia, Alzheimer's disease, and multiple sclerosis. And the nice thing is we are able to use these measures, what we are doing in the brain, and then try to predict what age do these patients have. We are having a very high, let's say, a coefficient for this particular subjects, and this can be used also to, for example, like I said here, big data, because we're using a lot of patients, and then also look at associative measures. That's why we call this association. So these measures can also be used for prediction of age, and this works not only in the healthy controls, but also in the patient groups. Now we looked into the aging biomarkers, as also in the introduction was talking about that we also built not only all these different platforms for electrophysiology and imaging, we also look into RNA analysis platforms. And here I show some examples that we do, of course, some pre-processing steps, and then we use, of course, several toolboxes, and then use the sequencing methods in order to sequence in a single cell level that what, in this case, spatial transcriptomics, meaning that we have tissue from the human brain, which is, of course, post-mortem, or we have the capability when we are operating these patients that we can get a certain tissue from certain part of the brain, and then look in that particular brain tissue, in the mice, in the human, what type of cells are present here. Just an example, for example, astrocytes or microglia, or even the neurons, and then map these specific transcriptomic signatures to the human brain, so that we not only stop at the electrophysiology or the imaging, we also look into these transcriptomics framework. And then what we achieve is that we try to use this platform development and then see what kind of cells are present, and then we can hopefully integrate this type of cell types also to the previous analysis methods and previous results what we have from these patients. And here I give you one single example in a again in a different mice or rat model. In this particular study, what we did is we looked at a specific gene in the kidney of these two type of mouse. This is one is called nanospelex and the other one is the rat. And why we look into this is because this nanospelex is then have a lifespan of greater than 20 years, and this let's say a type of mice is cancer resistant. So that's what we try to do is in this mice, how does it differentiate to a normal rat? So that is hypoxia sensitive, meaning that blood pressure, and have a shorter lifespan and not cancer resistant. And then we build an hypothesis that adaptation to hypoxia, because they are tolerant, when you reduce the oxygen in these nanosplex, they can have this live without this oxygen even less than 6%. And does this relate to the cancer resistance? And we looked into the one specific gene in this kidney of these rat and nanosplex, that's the ALDOP gene. And you can see the expressions are overexpressed in some parts and then also underexpressed in certain parts of these exons, just giving us the impression that in this rat, that it is kind of modulated for this cancer resistant, that you have certain exons directly get adjusted or adapted to this hypoxia, and that's why they are cancer resistant, compared to this rat, which is always very high, which is then not good, that's why they have not cancer resistant and have this shorter lifespan. So these are then again models which can help us use this for translating to the human. We have done this specifically now in the mice or rat and nanosplex, but we want to also transfer this to the human. And this is also, of course, not so easy, because you need to have, in this case, kidney information from the human so that you can do such type of analysis. So now I give you in a general overview what kind of platforms we are developing in our group. The first platform is then for network biomarkers. So again, always the problem comes that when you have this multimodal data, meaning that you get from the brain, from different modalities, several types of data sets. So all these are different type of data set. We looked into the diffusion tensor imaging. We also talked about the functional MRI, where we look at the activation, or we talked about the structural data, where we look at the volume of these cortical regions or other regions, or we can look at EEG, which we record electrophysiological activity, or we look at the aging, where we looked at hypometabolism. So all these modalities give different information, but the nice thing is you can look at individual network topology, meaning here as a circle shown with different colors, that how these different modalities are giving you this connectivity information between the regions, and use this for model training. And of course, the ultimate aim would be to either cluster, classify, or even predict the clinical score of patients in the initial diagnosis so that you can tell how do they progress in the disease after a certain time. And based on this, we can then look at translational research, meaning that not only stop at the human brain, which is shown here, for example, the MRI, or we can go into the basic research, meaning we translate this human data information, what we found, into the basic mice brain. And then, like we already talked about, the different recordings, what we get from the human, and look at certain resonant activity, how when we stimulate, what changes, and when we are very robust findings on these neural activity, then we can go to different states in the brain, meaning this is a very healthy state, that's the initial state, or you can modulate the brain by stimulation, and we call this neuromodulated state, or just a difference pathological means, for example, in the Parkinson's disease patients, what is the pathological state and how the brain regions react to this pathological state. So these are possibilities where we always try to translate from the human to the mice, and based on these network biomarkers, or we also have network changes which are then for precision medicine, meaning what is this network models useful for? They are useful for to have each and individual prediction for patients. It doesn't help us if we do this in a group. We need to have this information which is for each single patient. If we're able to predict, then gives us this nice possibility that we can look at their progression or even predict before how do they progress in their disease. So this is for the platform for network models. We also have a platform for omic biomarkers, where we, of course, try to integrate different omic data sets, which are, for example, probably have already seen before in other talks, like genome-wide association studies, where we looked at the genes, or we look at proteomics, or looking at molecular signaling, and then bringing this to, because these are high dimensional data sets, we need to reduce the dimension in a way so that we can look at network information. So that's why we call this a correlation matrix, meaning either it can be a correlation between proteins and proteins, or different gene expression to another gene expression in a certain region, let's say region in the brain. And then we identify differences in the network using, like I said, the clustering coefficient, network parameters, and then put this into a deep learning model so that you can then try to either classify the groups, meaning classify the patients to the healthy controls, or we can look at the distribution, how a certain group of patients are then different to the healthy controls. And then, like I said before, we also want to have information on the time, how do they progress, so we can have these correlation matrices, how do they develop over time, and then use this for our prediction models. And based on these platform for omic biomarkers and all the other platforms, we are then able to not only try to identify the different pathological information, how a diseased brain compared to the healthy brain, but we also want to find out how we can use this to predict the different states in the brain for each of these patient groups, for example, for the Parkinson's disease or multiple sclerosis, or in the aging group. So this is how we work in the group so that we can translate each and every particular finding from the human to the mouse, or back translation, we call it, from the mice to the human brain, because we have more detail in the mice brain compared to the human brain. And of course, all this is not possible without a good group of people. So here is the final slide. Here are the acknowledgement. Here is the group members. We have a very interdisciplinary group. We have engineers, we have doctors, we have biologists, we have informatician, sports scientists. So this is always possible only if you have an interdisciplinary group so that you can do all this work. And here are some funding organizations which is used to do all this research work. Thank you.
Thank you so much, Professor Muthuraman. It was really nice and clear talk. So I hope audience has been benefited a lot from this talk. So I look forward to the questions now. So I request the participants to put their questions in the chat box, or you can also raise your hand if you want to interact with Professor Muthuraman.
Good afternoon, sir. May I ask a few questions?
Sure.
Sir, first of all, thank you, sir, for enlightening us with your remarkable and engaging talk. Sir, my first question is what are the key challenges you face to translate your research from human to mouse model or mouse to human?
So always it's of course not easy to translate from the human to mice. The key challenges could be, for example, like I mentioned in one of the slides, we will not be able to get layer-wise information from the human brain because we have the capability of recording in a certain part in the brain, but we don't have possibility to put electrodes that we have like in the mice, that these layer-wise information. This, for example, is a big challenge. This is sometimes done in epileptic patients, that they have the possibility to do such an recording, but not in case of Parkinson, or because you need to, of course, really state why you want to do this. So that's why it's not easy to, for example, getting layer-wise information. That's one key challenge. And another challenge is that in the mice brain, it's of course, it's not, we can kind of sacrifice the mice for the human, for example, the human experiments, meaning that we can slice the brain of the mice and then get into these signatures of, let's say, transcriptomics or RNA sequencing. But in the human, there is a key challenge because that you cannot basically do this. You need to, of course, do this only in the post-mortem brains, which is available. Then you can do this kind of sequencing approach. Or if there is a capability in some of the labs where we can do this stimulation, and the patient, of course, probably only 5 to 10% of the patients allow that we can take a tissue which is about, let's say, a size of 10 cents. So this is then giving us the information of this signature. So this, I would say, two key challenges still we are facing: how to translate layer-wise, and also this transcriptomic signatures between human and mice.
Thank you so much, sir.
Welcome.
So Mahesh Kumar wants to also ask. I see his hand is raised. So you can go next, Mr. Mahesh Kumar.
Sorry, was there a question or?
I see someone has raised the hand. So I just was asking, requesting to ask his or her question.
So I see a question in the chat from Rajesh Anand. So how to confirm origin of biomarkers if we use the NMR? If you're talking about magnetic resonance imaging, like I said in the slides, that we divide the brain into specific regions, and based on this regions, whatever analysis what we do, for example, the volumes which I showed or activation maps, then we can know exactly origin. If you talk about the region itself, then we know then exactly from this region we find this difference between the human healthy subject and the patient cohort. Then we will know also the origin. If you talk about different modalities in the magnetic resonance imaging, then of course, for example, in the structural modality, like I said, we look at the thickness or the volume of this particular region and then know how different they are, because we always compare this to the healthy controls and then see how different. Or we look at, let's say, fMRI, then we functional magnetic resonance imaging, then we look at the bold response, meaning how does the blood in the particular part in the brain is differentiated between the healthy and the patient brain. So then we know exactly the origin of these biomarkers, let's say activity or connectivity. You're welcome.
Any other question? Okay, so maybe I can ask one last question. So how challenging do you find to integrate different modalities nowadays? Because people are looking at different kinds of modalities and try to integrate them. So what is your thought process, or maybe what is your opinion on what can be the best way to go ahead with?
That's a very good question. So I think that's a big challenge what we are still facing. Like I pointed out in the slides, we have these good concordance, let's say, between electrophysiology and neuroimaging, meaning like magnetic resonance imaging or other modalities in the magnetic resonance imaging and electrophysiology like the EEG. We have very good concordance, but when it comes to these modalities and then you want to integrate this with omics, then it's of course a huge problem, meaning how to integrate these two different modalities, because they have different, of course, type of data sets. And one way what we are currently trying is to reduce the dimension, like I said in the slides, that it's not possible to stay in the same dimension and then try to integrate them. We try to reduce the dimension either by correlation matrices or by independent component analysis, meaning we try to find clusters so that we reduce the data set of omics to a certain level, which then can be directly related to the, let's say, imaging modality or electrophysiology. And then I think one level lower is then basically this network connectivity values. So once if you have, because of course you know that you cannot get omics from the whole brain, you need to, of course, in the human, let's say, you're very specified to a certain part in the brain. So that's why it's also you need to get the similar information from the human brain so that you can directly correlate. So it's not, you always need another parameter or variable, like let's say a clinical score or a behavioral parameter. How do they do in a certain task in order to integrate or relate? There is, I think, very difficult to directly relate to these two regimes, neuroimaging, electrophysiology, and this omics. I think that's a big challenge which is still open. Yeah.
All right, thank you so much. And let me see if there are any other questions from participants before we close. Does anyone wants to ask any question?
Yes, sir, can I ask one more question?
Sure, sure.
Please, sure.
Yes, sir, can you talk more about like time series analysis for prediction of state of the disease in future? Like you talk about like correlation matrices and all. So any other prediction model that you're using in your lab?
So I think the time series is something very valuable, meaning you get more information about the brain from a current state. So the time series is then, of course, sometimes, like I showed in the slide, we use 256 channels, like in the head, those blue dots. So you have 256 time series from a certain time window. And what we do is we also record at a very high, very high frequency, meaning in 1 second of data, we get 1,000 data points from each of these EEG electrodes. So the good thing about it is once we have this, what we do is we try to locate the source or the origin of this activity from a certain oscillation, like I talked about, let's say delta. Then we can only do this with the time series. And another important predictor for us is this effective connectivity, meaning if you have two regions, how do they communicate between each other? Meaning you can show with the arrow that region 1 is giving information to region 2. This is only possible if you have the time series. If you don't have a time series, if you're talking about cortical volume or structural data, you cannot do this analysis because you don't know the time information to predict how one region is communicating to another region. So this is one important predictor for us when we use the time series, how two regions are communicating and which direction is this information going. This is giving you detailed information of the human brain or even in the mice.
Thank you so much, sir.
Thank you, Professor Muthuraman, once again. So we want to capture one snapshot so that it gives a kind of proof that you are here, we are here. So I would request also some members from panelists to turn on their videos if possible, so we can take one snapshot. Rajvi, Monica, maybe you can turn on your cameras if possible. Okay, so we are done. Thank you so much. And with this, we are I thank all the speakers for today. Dr. Prashant, Mr. Arpit, Professor Muthuraman, once again, and we are closing today's session. So we will tomorrow again start at 10 a.m. as per IST. We will be having 3 sessions tomorrow, very exciting sessions. First one is on Introduction to Oncogenomics by Dr. Abhishek Kumar. Then we have another topic topic on ruminating microRNAs by Dr. Ravi Shankar, who is principal scientist at ISBT. Then we will have an exciting session on matrix methods in data science by Dr. Dilpreet Kaur, who is mathematician at IIT Jodhpur. So thank you all the participants also for joining today. And with this, we would like to close. Thank you, Professor Muthu.
Thank you.
Bye.